Okay, I think we're going to get started. Hello, I want to extend a warm welcome to all, both on WebEx and in person. We're gathered to hear about the application of big data to pediatric safety studies. My name is Ann McMahon, and on behalf of the Office of Therapeutics, or OPT, at the FDA, Renan Bennell and I would like to invite you to listen and participate in our exciting conversations about big data in the healthcare setting. We would like to acknowledge all those people in OPT who have pushed this effort forward. We would also like to acknowledge the Division of Pediatric and Maternal Health in the Office of New Drugs at the FDA. This effort would not have been possible without their support. Before we start the workshop, I wanted to make a few brief points. The workshop is focused on big data in the healthcare sector. There are certainly many definitions of big data, even in the healthcare sector, and we focus mostly here on big data as it relates to observational healthcare data, especially in combined data sets. The first day will focus on big data in all the healthcare sector, and the second day will focus on the particular considerations of the use of big data in pediatric studies. This workshop will focus on the use of big data to assess medical product safety and not so much effectiveness, since tackling one of these areas is quite enough for a two-day workshop. We have made available to you a list of questions to be addressed to each panel. We will start by addressing these questions to the panel, followed by addressing these same questions to the audience, and thereafter open the floor to audience questions. We have an exciting group of speakers, and we'll now move on to the workshop. Our keynote speaker is Dr. Robert Califf, former commissioner of the FDA. Dr. Califf is Vice Chancellor for Health Data Science and Director of the Center for Integrated Health Data Science at Duke Health, the Donald F. Fortin, MD, Professor of Cardiology in the Duke University School of Medicine, and an advisor for Verily Life Sciences, a member of the Alphabet family of companies formed by Google. Dr. Califf.
Thanks, Ann, and it's great to be here with you today. A little bit nostalgic for me, I have to say. I miss the FDA a lot. It's a great organization, and it's fun to still be involved in these things. I look forward to the discussions over the next 2 days, and I think there's great potential in this field of big data, whatever it means, and I'm sure many of the speakers will fill in definitions. I took my charge to be just to give a very high-level overview of what's possible, and certainly should acknowledge that in my joint role in a university and with a company which has made its way in big data, that there are a lot of specific things I could talk about, but I'm not here to talk about any specific policies, but more the general overview of the field and the things that are possible. My first slide just talks about what I regard as the frontiers that need to be addressed. And when I think about big data applied to children, these are the areas where I think new focus should be applied. And I think about two dimensions. Most of this discussion in the next 2 days, as I see it, will be about large data sets in the healthcare delivery setting. But I also want to make the point today in several ways that the dimensions of data that can now be collected in real life at a much lower cost with a rapidly occurring reduction in cost make it possible to take a whole new look at human biology. And I particularly will emphasize several times maternal-fetal systems biology. I think is one of the most important areas that we can work in now, because we've been so limited by computation and the ability to measure that we're constantly making guesses about what to do in the circumstance of pregnancy. Obviously, evidence generation with pragmatic studies will be a big part of the discussion, but I believe this does get intertwined as we get into real life and real health systems with both implementation science. That is, how do you do research that has a lead-in to what could be done about the finding in a healthcare setting, and also beyond the healthcare setting into what really matters to most children and most people, which is the 99.5% of the time they spend not in a clinic and not in a hospital. So the social determinants of health are rising to the front in measurable ways that we used to just talk about or address through sampling methods, but now we can measure social determinants directly in large populations. Underlying all of this, I think, is a major set of issues where consenting adults can participate in this new datafied world, but we have special issues that are obvious with children about the ethics of participation, particularly with some of the things that I'll show you as I go through the talk. I just show this slide, which is an FDA slide that I borrowed from the current director of the Office of Pediatric Therapeutics, just to make the point that we're standing on the shoulders of a tremendous amount of work of people inside and outside of the FDA that have taken this field from something where guesswork was almost 100% of the effort about pediatric therapeutics to a situation where bolstered by laws and regulations, we now have a great impetus to do studies in children so that we have answers, so that parents can be assured that when practitioners make recommendations and apply therapies, it's being done in a rational way based on high quality evidence. And in many areas of policy, as I got to know it as FDA commissioner, it was easy for academics especially to talk about theory, but here we have real results to show that are quite dramatic and important. Documentation that not only were pediatricians right when they demanded that studies should be done in children, but that when the studies have been done, it's resulted in a change of the view of how medical products should be used in many of the cases. But this is only the beginning, and I think we're now entering a very different world in which we can expand these findings into the context in which therapies are applied in children. And I would argue that from an American perspective, there's never been a more urgent time, at least in my lifespan. And this does have to do a bit with lifespan. I borrowed this slide from Mark McClellan that he used to show when he was FDA commissioner. I still show it as a part of almost every talk, because, just to make the point that I'm actually an optimist despite everything else I might say in the course of the day, because my tendency is to focus on the problems, believing that things that are going well don't really need my attention. And so the point of this slide is just that since 1900, we've seen an improvement in survival in this country every year except 1918 until last year's data came in. But the point is the trajectory has been in the right direction. There's no reason why this shouldn't continue. People are living longer and doing better than ever before. And of note for this audience, in addition to cardiovascular disease, my specialty, the other area where the biggest difference has been made in life expectancy is care of pregnancy and low birth weight infants. But in the recent work by Chris Murray and his group in Seattle, there's some clear alarms that are bells that are ringing about the United States that we'd better pay attention to. This is a map of life expectancy in our country by county in the United States that appeared in JAMA just a few months ago. And you can see that we don't have homogeneous life expectancy in this country. And in fact, if you look at the map, it looks eerily similar to the election map in many ways. But this is true for almost every disease that's a major disease. And Murray and his group have now put out a whole series of papers about the big causes of death on cancer, heart disease, etc. And you can see major areas that are red, which means lower life expectancy. This is concerning because we have a lack of the benefit of the things that we're learning in some areas of the U.S. compared to others. But much more alarming is the trajectory of life expectancy shown here, in which we have many counties in the U.S. where we're seeing a reversal in life expectancy for the first time in a long time. And there are many papers just in the last month of people arguing about why this is the case. But it's obviously intertwined with a series of factors that have to do with culture, with use of healthcare, and with decisions that people are making. And, of course, in some areas, particularly in Appalachia, as the most outstanding area, you see the bright red there, the opioid crisis is exerting a huge impact on fetal maternal mortality and bad outcomes in general. Now, for those of you who are thinking about the broader use of data beyond the traditional experimental setting of regulated research, as I've made the point already, it's time to bring in the social determinants of health. And one of the great things about Murray's paper for a doctor like me is that, the question always comes up, does medical care matter at all after you've taken into account the social determinants? And the good news is that in their regression equation, medical care actually did matter. But it was a minority factor compared to where you live, what your income is, what your education is, and your race. So as we think about pediatric therapeutics and the situation in which therapeutic decisions are made, it's absolutely critical now that we have some of the new capacities that I'm going to talk about, that we really begin to think about how to integrate the holistic picture, not just the micro picture it might be seen in simple biological studies. This is also a standard slide set for me, but just to make a point that the capacity to use large populations now to generate evidence is growing very rapidly. We're in an accelerated phase now, which is tremendously exciting. And a lot of the accelerated phase is because of work done by people in the FDA and through collaboration with others, just to make the point that there is a rational way to think about therapeutics in the first place. This is not just random chance. That if we do the best early clinical trials, figure out what works, and then use large data sets to understand how to refine therapy and to understand the practicality of how to deliver that therapy and measure outcomes, we have a cycle that can lead to better and better outcomes over time. And this has been shown in a number of specialties as they've gotten onto this issue. I would argue it's been a lot harder to do in general practice, like general pediatrics or general medicine, because the doctor's office and the hospital are so heterogeneous that it's hard to pull together quality systems in a way that you can in a focus factory like my specialty of intensive care cardiology, where you have a place where people come in and you can run it more like a factory, actually, with quality metrics and a lot of attention to the details. But this joint increase in computation and the improvement in communication systems, I believe, is now making it possible to apply this cycle of therapeutics across the board to many areas of medicine. And it will take a change in our current system. Our current system is simply not configured to do the job. And this is demonstrated not only by the fact that most of the questions that rational parents might ask about their children are not currently answered. That is, our research system is simply not asking the questions that need to be asked, but also by the fact that practitioners have a hard time participating in the system because it's seen as onerous when it's put in place in the middle of a busy practice. The fact that practitioners have a hard time participating in the system because it's seen as onerous when it's put in place in the middle of a busy practice. And if you don't believe it, I'd urge you to go make rounds at our leading academic medical centers and talk to the doctors about how they feel about participating in research right now. It's really tough because there's tremendous pressure on efficiency in the system. And the fact that we've created sort of a parallel universe of clinical studies that demand that the studies be taken out of clinical context into a more experimental setting puts the practitioners in a very difficult position of not having the time to do it well by the current rules. And so we need a different system that takes off after the basic studies have been done to really understand how to use therapeutics, what the effects of drugs and devices and behavioral interventions are in real practice. And the great news here is that the organizational substrate is coming into place, and it's nothing new. It was envisioned by the traditional HMOs decades ago that when finally medicine caught up with the rest of the world and information became digitized, that we could treat medical clinics and hospitals much the same way that most successful industries run today in the United States and globally. That is, when you interact with the system, the product of that interaction is captured in digital form. The analysis of the information from that digital data is not just done locally, but is combined across places of interaction to yield sample sizes that can produce generalizable and valid results. And we're now in a phase where the interactions are being captured digitally. And as I'll show you, outside of the medical clinic and the hospital, the ability to use this information in real time on a massive scale is in play. Inside of the health system, unfortunately, we're still in the phase of entering a lot of data, but if you talk to practitioners, they're not getting a lot of information back. So this needs to be a focus. But a simple way to think about this is that, and this is a US-oriented talk, is that we're now in a phase of rapid consolidation. In half of my job at Duke University, I'm very aware of it because our competitor, UNC, of course, we love each other. We're only 8 miles apart, but there's a few areas of competition, sports and healthcare delivery. They just announced that they're combining with Carolina's healthcare, which is a $9 billion system. So combined, it's going to be a $14 billion healthcare system in North Carolina. And much like the HMOs have been doing for some time, particularly Kaiser, if you look at any of our systems now, we have a whole series of hospitals and clinics that are essentially under joint ownership with a tremendous need to ingest and analyze data to run the business of the healthcare system. And so data comes into a central source where it's analyzed. Every big healthcare system has a data warehouse now. These data warehouses are not getting worse. They're going to get better and better over time. But what's missing right now is the information going back out to the practitioners to improve care the way it should happen. But I have great reason to believe that this is going to change pretty radically over the next few years to be something that was originally envisioned. And thanks to the FDA and other parts of government working in public-private partnerships, I think national systems are now coming into place where it's hard to even imagine a sample size for a study that would be too big to be done in the United States alone. Now, as Patrick's going to talk about, there are a lot of other reasons to do international studies besides just to generate a bigger sample size. And I'm totally in favor of doing international studies. Every country that treats children should have an interest in doing studies in their country to understand the context of care and the biology of people that live in that country. But the time, I think, is coming to a close when we have to offshore research from the United States simply because we can't do it practically in the U.S. And of course, you know, the key start to this, I think, in terms of scalability was the Sentinel project where, as I learned, one of the great tricks at FDA is to figure out what needs to be done and then work with the outside community and Congress to get Congress to write a law that instructs the FDA to do something that it wanted to do in the first place. And this law that said you have to aggregate 100 million people's records for the purpose of drug safety was something that was really a law that was not like an admonition. It was something that needed to be done. And the result now is we have a functional system with something over 180 million people's records in it and over 40 million under active surveillance in the system being used by the FDA, putting together claims data curated to a common data model. So this is a major resource, very pertinent to children. And I was very pleased during my time at the FDA to do a lot of work with CDRH. And I think the device part of the FDA is taking this to a new level by saying, and let me just say, I think it was wise what the FDA did to start, which is to sort of have a command and control system called Sentinel that was kept under the radar for the most part until it really got off the ground. Now it's really in a new phase, and through Reagan-Udall, it's being made available to the outside community. But the idea in the device part of the FDA was, let's create a relatively democratized network of networks that moves the device industry from doing one-off studies to doing studies that are embedded in registries about devices so that we have real-world data right from the start. And you'll notice the picture looks the same. The little clusters here would be individual health systems under the same ownership, which have a reason to collect data and use it for business and quality of care purposes. And by combining data across those health systems, we now have a scalable system with a sample size which is not constrained by the typical environment of a typical old-fashioned research site the way it used to exist. And this is a CDRH slide which I think pretty much tells the whole story. As we move the system from passive surveillance, depending on people to recognize a problem and then have the energy to report it, which is, again, if you make rounds, you'd say there's just no way we're getting anywhere near the reports of things that are really occurring. And even recently, in some things I've had a chance to write an editorial about, all it takes is one news story and suddenly the whole system's distorted by people reporting things that always existed, but they didn't have the energy to report before. But now we go to a system which is no longer passive and one-off, but one in which there's constant active surveillance where you can calculate numerators and denominators embedded in the healthcare system and where clinical trials are leveraging this information. Now this is, a work in progress. It's not fully formed, but it's where the system needs to go, I think, both for adults and for children. And recently, since coming out of the FDA, I was excited to go back to something I had worked on before coming to FDA, the formation of PCORNet. And I was asked to chair the board for the new not-for-profit, which is the emergence of PCORNet from a purely PCORI organization to a national organization to do comparative effectiveness research. But you'll notice, whether we're talking about drugs, devices, or comparative effectiveness, the basic system and the scalability of the system looks the same. It's built on healthcare systems, organized to collect information digitally, and then working across those systems using common data models to have data, and importantly, engaging clinicians to do prospective studies. And on that note, the NIH has been playing a major role. Another project I was involved in before going to FDA, which is moving along quite well without me, is the NIH Collaboratory, which is now engaged in 10 clinical trials in the post-market phase, being done at about 1/10 the cost of a traditional regulated clinical trial. There's a lot being learned in this exercise. It's meant as sort of a way to break the ice in moving to clinical trials in which existing digital data is used as a basis for the trial so that you don't have to create this parallel universe of data that's used in the traditional regulated clinical trial. And there's a new round of trials about to be funded. The program, I think, has been deemed successful. And a number of the NIH institutes are now coming out with their own RFAs for similar kinds of studies. Just a quick word about PCORNet, since I'm very involved in it. We call it PCORNet 2.0. And the idea here is that by combining numerous health systems and patient-powered groups, we can do research at a much lower cost, at a much larger scale, and much faster. And just to give you an idea of how we see it coming together, if you take health systems with their data warehouses for both claims data and electronic health record data, and combine that with patient groups, including the registries that they bring with them, which are proliferating, and I might add, with great help now from FDA, particularly in orphan diseases, you now potentially have a national network that can deliver the goods at a low cost. And if you look at the upper right-hand corner here, you'll see Improved Care Now, which is an example of a major pediatric-based effort that's part of this network. And within the health systems, there are 13 groups of health systems, but altogether 34 health systems that are combined into this effort. And you'll notice PedsNet, again, in the upper right-hand corner, is a major part of this effort focused on children. So if you put all this together, currently we're curating once a quarter to a common data model information on 122 million Americans. So it's over a third of America now is in this network. And it's being linked with the practitioners so that we not only can do passive observational studies, but also prospective clinical trials and prospective observational studies when needed. Then just to make the point that policy is supporting this evolution, something I think that's part of the reason to have this meeting. Written into the Cures legislation and the user fee agreements is tremendous support to help industry get over the regulatory inertia that we all know exists. And all of you know that without legislation, it would have been hard to get those studies that led to changing the labels in drugs for children. In the same way, I think this legislation, in a very nice way, is pushing the FDA and industry with help from the rest of the ecosystem to work together to figure out how to use evidence in the real world in the best possible way. And in this FDA slide, the point is made that the sweet spot right now is in the post-market phase, but I do want to make the point that throughout the entire continuum, even going back to basic biology, the use of big data and the increasing using of digital phenotyping, which I'll talk about in a second, will turn out to be very important. And then if there's one point I want to make in terms of thinking about this conceptually, I would refer you to the article that we wrote just at the end of last year with Rachel Sherman as the first author. The point being that people tend to talk about real-world evidence and randomization as if they're polar ends of a spectrum. In fact, they're not even in the same spectrum. They're two different dimensions. One is, where does your data come from? It might come from a special research clinic set aside from clinical practice, or it might come from a regular clinic or hospital. Increasingly, it may come from people's homes or their environment as they're walking around in real life. The other dimension is, what's the method that you're using to design your study and analyze the data? And that could include randomization, even if it's real-world data. And in fact, the argument at the FDA that we made is that the best, highest form of the art of clinical research is going to be randomization in the real world. And I'll say a bit more about that in just a second. So deeper information about mechanism is going to be available soon. And here, I do want to acknowledge the conflict that I have, that I work for the company which is sponsoring this study, and I also have an academic appointment at the two academic centers that are enrolling patients in the study, so I couldn't be more conflicted when I discuss this, but it's really just to make the point that the way we used to think about data in general is undergoing a radical change due to the changes in computation and the ability to align processors on a global basis for just-in-time purpose. So things, I'll have to say in my new life at Google half-time, I have not seen a problem yet where the engineers said the size of the data is an issue. I just think about that for a minute. For someone from a university background, a lot of the work I used to have to put in was figuring out how to pay for storing the data. This is undergoing a radical change. And we used to be limited in what we could measure just because of the cost. But the cost is coming down at such a rate that the study that we've just embarked on, called Baseline, is measuring everything we can possibly measure about 10,000 people. This is just a list of the studies that every person is having. But it's not just a traditional in the clinic evaluation. People are going home wearing devices and having sensors in their home that also measure things 24/7 and stream them to the cloud. This is no longer a foreign concept. If you wear an Apple Watch, for example, this is the way more and more people are living. And it's not just the computation that's important, it's the ability to interact with the person in real time in a way that allows the recording of things like patient-reported outcomes in real time in an almost passive manner that's really second nature. So if you look at the dominant cell phones today, the things that are measured passively are voluminous, and the ability to interact with people because they're doing it anyway. The average person in the U.S., as you know, touches his or her cell phone every 17 seconds. So this is just a part of everyday life now. So if you look at just the clinic visits involved in this study, every clinic visit captures 6 terabytes of data. And it's hard to even get your brain around the size of the data that's being ingested in the study. And that doesn't count the fact that I'm wearing actually the Apple and the Google Watch, so as not to appear biased here, but the study watch I'm wearing measures 17 parameters continuously and downloads them whenever you want to download them for analysis. So that's big data. What are we going to do with it? And I want to argue again that maternal fetal health is probably the place where the greatest progress can be made, because we were so limited up until now about what we could measure and the way we could measure it. And I'm not going to dwell on the details here because I'm not an expert in the topic, but as I've looked at it, understanding, particularly for things like drugs and intrauterine devices, the impact of interventions, the concentrations of drugs, the effects across the placenta, very difficult. But using modeling and the increasing ability to digitize information and measure it, I believe we're about to enter a new era where the systems biology of both involved organisms can be measured, and models will tell us a lot about how to dose drugs in ways and understand what's happening not just to the mother, but also to the fetus. And this is just to show that at least early work, I think, is showing that predictions, when backed up by real measurements, can work, at least in the simplest form of this. And many of you in the audience are expert in this arena. So then finally, I'm going to just rush through a couple of slides about the other part of this, which is probably not part of the meeting otherwise, and you may be a little bit entertained by it or frightened by it, I don't know, depending on your perspective. Because it's not just the computation that pertains to real-world evidence, it's also the ability to interact with a person that's important. It now becomes critical for people like the FDA and the rest of us as scientists to think about not just are we doing the research, but how are we transmitting the information in a way that leads people to truth and expertise. And I don't need to tell you that the very tools that I'm talking about can be used either for really good purposes or really bad purposes. People are confused but now about what truth and expertise are. And I do want you to know about what's going on with Google search. It's not the part of Google I'm working in, but I have been allowed to have interactions with the search team. But it should give you an idea, and I hope inspiration as you think about scalability of real-world evidence for what could happen. Right now, 3.5 billion people every day do a Google search. Is there anybody in the room who hasn't done a Google search in the last week? So, and 1 of 20 searches are health-related. So it's over half the world is doing a Google search every day. And the mission of the health part of Google search is to make health information universally accessible and useful. But people are searching for a reason, often that they don't want to have to go to their doctor. If you actually look at what's behind a large percentage of Google searches, It's either they went to the doctor and didn't understand anything that was said, or they're searching for a home remedy to avoid having to get into the healthcare system. And what we need to be able to do as you work through real-world evidence is not just generate the evidence, but direct people to reliable evidence, because increasingly they're going to be making their own choices by interacting with things on the internet that can direct them to good or bad. So how do we do this? And I'm just going to show you what's happening with depression, because it's just the initial foray into this. First of all, if you're searching Google now and you're indicating you're thinking of committing suicide, rather than getting a YouTube video on how to commit suicide, which was a possibility 10 years ago, you now get directed to the hotline closest to you for suicide prevention. But as you know, 10% of America is depressed, really 10% in every part of the world, and search is available in 110 languages now, with a huge impact. And it looks like the average person who's indicating that they may be depressed is not getting to medical care for about 7 years. And so there's a real interesting question here. How do we help as people are searching, not to tell them the answers, because people don't want to be told by Big Brother what the answer is, but to direct them to truth and expertise so that they can help themselves. And if you think about 20% of 3.5 billion searches being health, 10% of people being depressed, this is a very large number of searches that are going on. So 2 weeks ago, the team turned on something that if you're searching and it appears that you're depressed, a question pops up that says, would you like to take a questionnaire? And this is a PHQ-9, which is a standard questionnaire used in clinics for depression. And if you score high, the information that you get looks different than if you're not depressed. And you get referred to a patient advocacy group that deals with depression. This begins to give you an idea of scalability on a global basis. And I would urge you as you go through the day not to just think about what's possible now, but begin to think about, if the cultural barriers were broken down, how rapidly you could accrue knowledge. But in order to do this, we need to think about both the ethics and the education of people of how to use these tools appropriately. And it's critical that we spend considerable time in the system that I described, making the data better so that we can get better answers, and that we develop a culture of sharing in science, which I would argue is the major problem now. That is, people have shown in other areas that they're willing to share massive amounts of information if it will get them what they want. And who doesn't want the right dose of a drug or the right interventional technique for their child? But our culture of science is impeding the transmission of information and the sharing of it in a way that can answer the kinds of questions that you have. So I'll finish with this depiction of what does happen with Google search, and ask the question, why doesn't this happen in medicine? And I'm sure you'll answer it in the next day and a half. The point is, 3.5 billion people searching every day. Every time a search is done, the information is used, I can't say in real time, but pretty close to it, to learn about what people are asking, to understand the use of language, to understand the context of the question, because often people, when they type, they can't, just like when they talk, they're not precise in what they're trying to ask. So you want to help them know the answer. And the way this happens is the use of machine learning and other quantitative techniques. But when there's a question about how to approach things better, the standard is not to make a guess. The standard is to do what's called an A-B comparison. The A-B comparison is a pragmatic randomized trial. Is A better than B? And this is done routinely many, many times a day in the Google environment, because it's known that you can't get the right answer just by observing. You often need randomization to clarify the causal inference. And what's really fascinating about this to me is that this is a part of business. It's really not being done for healthcare. But if you apply this kind of thinking to how do you get the right dose of a drug, how do you understand when the use of a therapy is appropriate, the rate of learning, if you had all the pediatricians in the world, or half, contributing to the knowledge base, I think would be several orders of magnitude faster. So I hope that this meeting will be a sentinel meeting in moving the field forward, that we'll get into a phase where on behalf of children, we're all working together to use this digital data, which is now uniformly available, to bring parents and their children the best treatment possible. Thank you.
Thank you, Dr. Kayla, for a really inspiring talk, start for this workshop. Next is Dr. Mona Khurana, who is a pediatric nephrologist who joined the FDA in 2009 after a 5-year academic career at Children's National Medical Center. Dr. Khurana is currently a pediatric team leader in the Division of Pediatric and Maternal Health. She will be talking about regulatory context, big data, and pediatrics at the Food and Drug Administration. Thank you, Dr. Khurana.
Good morning. It's really a pleasure to be here. I'm looking forward to the discussion over the next 2 days to learn how real-world data can be structured and analyzed to provide real-world evidence of drug safety, and how this evidence could be leveraged to help inform FDA's safety assessments of pediatric products throughout their entire life cycle, from marketing application through approval and in the post-marketing setting. I have no disclosures, and this is the outline for my presentation. I thought it would be useful for today's discussion to describe the progress we've made so far with promoting pediatric drug development, the challenges we continue to face, the safety considerations unique to pediatrics that we currently think about during the life cycle of product development, and the potential ways real-world evidence could be used throughout this life cycle to help improve the efficiency of pediatric drug development. Because this is a pediatric workshop, I thought I should begin with how we define the pediatric population. FDA does not actually have a standard regulatory definition. Our regulations define the pediatric age range differently based on the context of use. Labeling regulations for prescription drugs and biologics define pediatrics as birth to 16 years of age inclusive. Regulations for medical devices define the upper pediatric age limit at 21 years. For purposes of clinical trial enrollment in the U.S., our ethics regulations consider children to be persons who have not attained the legal age for consent to investigative treatments or procedures. And this legal age varies depending on the laws of the individual states where U.S. enrollment sites are located. So prior to passage of the first incentive provisions to complete pediatric studies in 1997 under the FDA Modernization Act, more than 80% of approved drugs had no pediatric-specific labeling, and pediatric practitioners were faced with either one of two options. Either not treat children with potentially safe and beneficial drugs because they were not approved or studied for pediatric use, or to use them off-label based on adult studies and limited pediatric anecdotal experience. Publication of the 2000 ICH E11 guidance was an important milestone for pediatric drug development because it marked the first time that global regulatory bodies collaborated to establish general principles for how drugs should be developed in children. The guidance emphasizes that the best therapy for pediatric patients is approved therapy, and that the safety and efficacy of drugs in development should be studied and understood in pediatric patients if those drugs are anticipated to be used in the pediatric population. An addendum to this guidance is hot off the press and was just published last week. The addendum is intended to update the 2000 guidance by providing clarification and regulatory perspectives on current and evolving topics in global pediatric drug development, including ethical considerations, age classification and pediatric subgroups, including neonates, pediatric formulation development, efficacy extrapolation, and modeling and simulation. Despite the general governing principles outlined by ICH in 2000, advances in pediatric drug development in the U.S. still fell short until enactment of the Best Pharmaceuticals for Children Act in 2002 and the Pediatric Research Equity Act in 2003. The incentive provisions in BPCA and the requirement provisions in PREA share the common goal of conducting pediatric studies for the purpose of increasing pediatric-specific product labeling information in prescription drugs and biological products. Passage of both landmark pieces of legislation has led to 650 of the more than 700 labeling changes FDA has approved over the last 19 years to incorporate pediatric use information in labeling. But we've still had challenges moving into the 21st century. One big challenge we continue to face is the long lag time between adult approval of a product and subsequent pediatric approval or incorporation of pediatric use information in labeling for that product. We recently reviewed 649 pediatric labeling changes approved by FDA since 1998 and found the mean time to incorporation of pediatric use information in labeling was between 5 to 6 years. There's widespread variability in how long pediatric approval may take, depending on the class of drug and the indication for use. Some drug development programs have taken up to 20 years. Another common challenge is the practicality of conducting clinical trials, particularly for pediatric onset diseases which occur in only a small segment of the pediatric population. Because of the limited size and duration of pediatric safety studies during product development, detection of adverse reactions is unlikely until there's more widespread use of the product post-approval. Establishing the long-term safety of a product intended to be used in some cases for the lifetime of a pediatric patient is particularly challenging to do in a prospective fashion. Although BPCA and PREA have given sponsors incentives and requirements to conduct pediatric studies, neither law has made it easier to get these studies done. Both laws still require pediatric studies to be rigorous enough to meet the same evidentiary standard for drug approval as in adults. So what are the safety considerations unique to pediatrics that we currently think about in the life cycle of a product? Several key issues must first be addressed to determine if pediatric patients can even be safely enrolled in clinical trials. These issues are listed on this slide, and I'll describe each one and how they are addressed within our current regulatory framework in the next few slides. There are two important ethical questions to ask at the onset of any pediatric program. First, can the public health objectives be met if study enrollment is limited to adults? If not, how can we maximize the applicability of existing adult data to determine what pediatric studies are really needed to support product approval? If pediatric clinical studies are needed and involve more than minimal risk, is there enough evidence that the product has the potential to provide direct clinical benefit to individually enrolled pediatric patients to justify exposing them to those risks? This is known as proof of concept. Importantly, proof of concept is not the same as approval. And lack of proof of concept could stall a pediatric program for a product which is a new molecular entity. Non-clinical studies are usually conducted to support first-in-human dosing and safety monitoring in adults and are an important part of the human risk assessment for drugs in development. Pediatric enrollment in clinical trials generally proceeds after there's been considerable experience in the adult population. And the need for juvenile animal studies prior to enrolling pediatric patients is considered on a case-by-case basis. Pediatric dosing is generally based on PK studies designed to determine how the dosage regimen should be adjusted in pediatric patients to achieve a similar level of systemic exposure that has been found to be effective in adults. Dedicated PK studies may not be needed in all pediatric age groups, particularly in adolescents, if growth and developmental changes don't create substantial changes in the PK parameters of a drug. Because dedicated PK studies have conventionally been done with intensive blood sampling in pediatric patients, alternative study designs are being increasingly implemented, which rely on sparse PK sampling to evaluate drug disposition. Once a determination has been made that pediatric patients can be safely enrolled in clinical studies, then the adequacy of the collected clinical safety data becomes an important component of a product's safety assessment pre-approval. The safety database should ideally be able to adequately capture adverse reactions at the proposed dosage for the anticipated duration of use. Examining more than one dose level would allow better characterization of the relationship between drug exposure and resulting clinical benefit and risk. The database should include a sufficiently diverse population that could be reasonably expected to represent the anticipated target population post-approval. The size of the database needed to accurately capture relevant safety information depends on the size of the affected population and the frequency of the adverse reaction. Too often, these pre-marketing databases fall short. They cannot detect rare adverse reactions because of the limited number of pediatric patients with a disease of interest who are available for trial participation. The small study population often encountered in the pre-market setting raises questions as to whether the safety data are representative of product use in broader populations post-approval. Pre-marketing studies may also be too short in duration to accurately capture potential adverse effects with long-term use of a drug. And because the study population is already small, conducting a prospective comparative safety study when needed can be challenging. For all of these reasons, the short and particularly the long-term safety review of a product continues post-approval. FDA is congressionally mandated to review all adverse event reports for 18 months after any approved pediatric labeling change for a product and to present the findings to a pediatric advisory committee. This effort provides a window as to how these products are actually being used in pediatric patients in a real-world setting. FDA's post-marketing safety reviews are primarily informed by reviews of adverse events reported to the FAERS database. Data mining and FDA Sentinel Initiative are two other approaches currently used by the agency to identify safety signals that conventional pharmacovigilance approaches are unable to detect. The Sentinel system is a national electronic system FDA launched in May 2008 to securely access healthcare data in order to track the safety of drugs and other FDA-regulated medical products. And FDA systematically examines databases using statistical analyses referred to as data mining to identify unusual or unexpected product-related adverse events warranting further evaluation. The data capabilities and challenges with using these post-marketing safety databases were discussed at some length at last year's workshop, which focused on long-term pediatric safety studies. So how can we use real-world evidence to help us during the drug development life cycle in ways that will improve the efficiency of resources and provide information to support short and particularly long-term safety of products for pediatric use? Could the rapid and efficient use of real-world evidence result in decreased overall mean time between adult and pediatric approval? Importantly, from an ethical perspective, could we harness real-world evidence in such a way so that children are not unnecessarily enrolled in clinical trials? I'll try to touch on these questions in my last few slides. Real-world evidence could potentially be used to impact multiple areas in the life cycle of a product. It could be used to support the acceptability of extrapolating efficacy from adults to pediatric patients by providing evidence that the course of a disease is similar between the two populations. It could be used to support the proof of concept needed to justify enrollment of pediatric patients in clinical trials. Proof of concept is generally established by mechanistic studies in animal models and in vivo studies in adult humans during early phase development, but real-world evidence could provide valuable safety information from other drugs in the same class to inform this assessment. It could be used to help inform the need for juvenile toxicity studies, for instance, by identifying adverse reactions associated with pediatric use of a marketed, unapproved drug product now seeking FDA approval, for which the exposure-toxicity relationship needs to be further elucidated. Real-world evidence could also be used to identify clinical areas where opportunistic PK studies may be feasible. These types of studies are being increasingly used to enhance pediatric enrollment and reduce patient and caregiver burden because they allow collection of PK samples for a drug of interest that's already being used as standard of care. Application of large data sets could also lead us in the right direction if we're concerned about a potential safety signal in pediatric patients. The goal of post-marketing safety surveillance is to identify any previously unrecognized, unlabeled, serious adverse reactions. Real-world evidence offers this possibility by potentially allowing product safety to be captured in a broader population than in whom the product was developed. Finally, real-world evidence is perhaps most uniquely suited to capturing valuable long-term safety data in pediatric patients to help us better understand the safety of a product taken chronically during important stages in a child's development, and for adverse reactions which may not manifest until many months or years after drug exposure. Thanks so much for your attention. I'd like to acknowledge Lynn Yao, John Alexander, and Harry Sachs for helping me with this presentation. Thank you.
Thank you very much, Dr. Khurana. The next is Dr. Alison Cave, who is from the European Medicines Agency and has been leading the effort on big data collection and analysis. And she is here to talk about a workshop that the EMA put on big data and healthcare, the findings and conclusions of this workshop. Look forward to hearing this.
So, sorry, many thanks, and thank you for the invitation, Anne, and for the opportunity to provide some reflections on our workshop that we held last year. So this is my disclaimer. And first, to orientate you, I'm going to start by discussing why now. Why is now the time to start the conversation, mostly from the regulatory perspective? And I'm going to provide you some examples across the product life cycle, which I think illustrate some of the challenges. I'm then going to reflect on the key messages and themes of the workshop, which I think are very pertinent to the discussions today, and also very briefly describe a new regulatory initiative that's ongoing in Europe to try and address some challenges before finishing with some short conclusions, and I hope to not cut into your coffee break. So why now? Are we just tempted really by the law of big data, by everything that may be available, and is that why we're considering it? Or are there specific regulatory challenges which are beginning to present to us, and this may be even more pertinent to the pediatric and pediatric medicines where it's extremely complex, that are really demanding new approaches? And obviously I think the answer is yes, and I just want to highlight some sort of good key illustrations. So firstly, the scientific landscape is radically changed. If I think back to when I started my PhD, I was still going to the library to flick through current contents, to look for my PubMed, to look for my literature searches, which is just unbelievable to students today. But seriously, the explosion in technology is really creating new challenges in the regulatory context. And a really key example is obviously genomics. And we have an increasing number of medicines with either a genomic mechanism of action and or genomic biomarkers which are selecting the patient population. And this enables smaller, more focused RCTs, but it creates other challenges for us. And here, I think, is a nice case study. It's provided by Ehr Data, which is mutation modulating drug for cystic fibrosis. So cystic fibrosis is a life-limiting multi-system disorder, and it's caused by one of nearly 2,000 mutations in the CF transmembrane conductance regulatory gene. And sadly for CF, there's no definitive cure at the moment. Medications manage symptoms, but generally don't affect the progression of the disease. So Ivacaftor is a new drug which is developed in silico, which targets the G551D mutation in the CFTR gene, which is responsible for 4% of CF cases. So small percentages, not the predominant gene, but it alters the open probability of the channel. But in pivotal trials, Ivacaftor delivered an increase in FEV1 of 10%, which is great. But obviously, we don't know what the long-term safety is in this population, and that may be in a population, as was highlighted earlier, where patients are treated from birth across all development phases for their entire life. So clearly, here we need long-term studies to monitor the long-term safety of the medication. But because the point I wanted to make is because the medication is known, because the mechanism of action is known, the indication of Ivacaftor has been gradually expanded over following years to, and at last count now, covers 23 mutations. But this is a rare and small and difficult population to study, and the indication has predominantly been expanded on the basis of lab data coupled with clinical data rather than new RCTs. So I think the question is, in the future, the challenge is going to be determining the level of evidence that we require as a regulatory community to extend indications of medicines when further mutations are identified, potentially through next-gen sequencing. So I think this is a key point that we need to consider. The other challenges in innovative medicines and personal prescribing, i.e. patient stratification, which creates regulatory challenges. And here I talk also, here again, genetics driving patient selection. And the example I've used is genetics, but it could equally be a circulating proteomic biomarker or a metabolic biomarker. So when the genetic biomarker driving selection is strongly predictive, we can be more confident about, for example, prescribing a medication. And here, in a very well-known example of Angelina Jolie Pitt, who had a BRCA1 mutation, which gave her an 87% chance of breast cancer or 50% risk of ovarian cancer. This makes the decision-making process easy, be it here, well, relatively easy, be it here to have radical surgery or alternatively in other scenarios to prescribe a medicine which potentially has adverse drug reactions. But for other diseases where the risk is less predictive, for example, in Alzheimer's or in dementia here, that's the example I use, where your genetic risk or your family history may increase your risk of dementia by from 10 to 20%, that makes the decision much easier to potentially identify patients to be treated prophylactically, which could be 10 or 20 or more years in advance. And how do you assess the benefit-risk profile in those cases? And I think that's pertinent to children, but also many other populations. Again, rare diseases, very relevant to children, may be associated with more limited information at authorization, which creates regulatory challenges. And here I use a recent gene therapy product, Stremvelis, which is a corrective treatment for children with SCID ADH, very rare condition, prevalence about 15 patients a year in Europe. And it's indicated for those children in whom no matched donor was available and alternative treatments had not worked or available. And here the pivotal trial was in only 12 patients, and it was an open label trial. Primary outcome was 3-year survival, and in fact you can see that the treatment was very effective, with 12 out of 12, 100% survival at the 3-year time point. And this is in a condition that without treatment, the disorder is fatal. But here, again, what were the uncertainties? And here, the long-term durability of benefit was uncertain in comparison with sort of standard stem cell transplantation. Late failure, the need for further treatment, late toxicities, or long-term immunogenicity. And you can see that there were some autoimmune adverse events. So here, the treatment got a conditional MA, which requires data and data sources to follow up. And here, and you can see here, I make the point that we are seeing an increasing number of applications requesting conditional marketing authorization, all of whom are associated with post-marketing obligations and the need for data collection. And this is not a new point, but of course the unknown generability of RCT results to normal clinical practice really demands new approaches to gather complementary evidence. And here I show what I think is a very nice illustration, which was provided by Hapskit, who developed a propensity score model that predicted participation in either a randomized trial, which is shown here in red, or a real-world study, which is shown here in black. And in this case, it was looking at outcomes for non-small cell lung cancer patients treated with platinum-based chemotherapy. But that's not the point. It could be any trial, and this is actually replicated over many disease areas. But you can see that given a common total baseline characteristics, the overlap between the two cohorts was minimal. And this creates problems in translating benefits into the real world. Additional data sources are therefore definitely needed to better monitor risk-benefit in high-risk groups, those often excluded from clinical trials, and that's the case for pediatrics. And even in pediatric epidemiological studies, only a very small percentage are carried out in pediatric populations. And of course, it's the same story for geriatric patients. And lastly, an increasing interest in combination therapies to treat complex diseases also creates regulatory challenges. I show some examples here, but it highlights questions about when do we stop therapy? How do we determine dose-response relationships? Even more difficult in children. How do we understand ADRs, which only arise with the combination and not when used in isolation? And how do we handle or monitor drop-off in efficacy or the development of resistance when we can't titrate fixed-dose combinations? So it was on this background, and of course on the background of IT developments and technological changes which allow and open possibilities of access to data, that we decided to hold the meeting last year on identifying opportunities for big data in medicines development and regulatory science. This was in November of last year, and we really started by defining the big data landscape from a regulatory perspective in order to clarify opportunities and challenges. And we really wanted to arrive at what is needed for us to exploit this data in the regulatory context. So some key messages. First of all, we wanted to define the big data landscape. And the meeting considered all types of big data, a slightly broader scope. But I think the real thing we wanted to do was, first of all, think about how we might meet big data in the regulatory context. And we defined big data as data sets that's so large and complex that traditional data processing applications were inadequate. But thinking about how we might meet this data in the context of medicines regulation, we could meet it as data in large amounts or of a complex nature, reaching us in the margins of the more traditional analyzed and structured data. So it's, sorry, I've lost my slide set there, complementary. It's going to sleep. Really bored with the talk. So I'll give it a minute. It's quite hard to go on without any slide set. So shall I flick through? I don't have anything on my screen here. Okay, so can you go on, please? Quite a few. Carry on, yeah, please. I'm not sure which number it was. Continue. Yeah, one more. There, okay. Or we could meet data. The data may be actually slightly hidden and underlying the regulatory submissions, but it would be crucial to understand their presence and the robustness by which they were generated in order to make a competent evaluation of the submission as the whole. Next slide, please. So the data landscape, what did we consider? Next slide. And we considered the whole spectrum, and that's sort of a slightly different to this conference, but we really considered clinical databases, but also more the future, the social media, the mHealth, and mostly we were trying to understand how this data could be integrated with the more omics type data that's coming online. Next slide, please. So what are the challenges? Sorry, next, please. I think the first challenge is the speed with which the data is accumulating, and Rob touched on this earlier, but it's quite daunting to realize that 90% of the world's data has been created in the last 2 years. Next. And it's the frequency of which it's doubling. So every 2 months, every 2 years, the electronic healthcare data doubles. Next slide. And it's not going to get any better. So 75% of patients are expected to use digital health services in the future. So how do we future-proof our regulatory system to deal with this? Next slide, please. And this also touches on what Rob talked about. Really, what are the opportunities? And this was quite a startling sort of slide for me that was presented, that when we consider the data which impacts on how patients respond to benefit risk, it's estimated that 10% of that is clinical factors, and 20% of it is genomic. But 70% of how we respond to a medicine is actually influenced by our social, environmental, and behavioral factors. And at the moment, in the regulatory context, we have no handle on this at all. So we can't get that holistic picture and integrate this information to understand how patients may respond to a medicine. Next slide, please. So in terms of what we really focused on, we focused on data accessibility and integration. How will we access the data and what are the challenges? Next, please. And we will hear later today from Sentinel, we'll hear from Odyssey, and there's also other distributed data networks. Next, please. But it's complex to do these distributed data networks. It's complex to bring them together. Data is often siloed at individual centers. It's hard to access and it's hard to use. Next. And as a result of this siloed nature, productivity tools, especially IT tools, have been built for individual local usage, focusing on local data analytical solutions. Next, please. And we need to start to move towards centralized IT solutions, which will enable us to store the data safely and securely, so that's key, but also then enable these machine learning solutions to give us these insights that we're seeking. Next, please. But bringing this data together is very, very hard, especially if we're considering how we can integrate multiple different types of data. The data needs to be standardized, it needs to be structured, and it needs to be stored together to deliver that insight. Next, please. But we need to be able to find it. And there was a startling statistic that 80% of data that's generated today is actually not findable. It's not findable by electronic solutions. And if we are to avoid duplication of effort, massive duplication of effort that's generated in part by the scientific culture, but in part by how we store the data, we need to make data fair. We need to make it findable, accessible, interoperable, and reusable. Next, please. And I like this very much in that in the future, the world's most valuable resource is no longer going to be oil but data. Next, please. But we shouldn't only consider data. We shouldn't only consider collection of data. We really need to consider the clinical utility of the data that we collect. Next, please. And the point was made that data collection should be targeted. We should be collecting data that starts by asking the right question. We need to target our data collection. Next, please. And then we will collect data that will respond to questions that will translate into public health benefit. And we had a nice schematic, next please, from a lady from London Transport, who told us that she always thinks about data collection, that as a, whatever you are, be a regulator, be a scientist, you need to collect data that will give you insights to do what? So that you can make a decision your job expects you to make. So really moving towards targeted data collection that answers specific questions. Next, please. And actually, we had a nice illustration of clinical utility provided by Sir Munir Pir Mohammed at the meeting, where he highlighted now that 15% of EMA medicines, centrally approved since 1995, contained pharmacogenomic information in the label, and the vast majority of these was actually within in oncology. But it's an increasing trend, with 11% of medicines you can see on the right-hand side authorized in 2014, including pharmacogenomic information. But how much information do you need to actually get this drug biomarker information into the label? Next slide, please. And this study highlighted that actually for the majority of these drug biomarker combinations, the labels that they looked at did not have clinical utility within the evidence that underpinned it. And you can see here that actually only a third contained data supporting convincing utility. So they highlighted, the authors highlighted, it may be premature to include biomarker testing recommendations in drug labels when convincing data that link testing to patient outcomes do not exist. But I think this is a problem that is only going to expand as we get more and more biomarkers and move to panels of biomarkers and ultimately to next-gen sequences. And the regulation of these biomarker panels will be very challenging, as will be the decision about what to put in the label. Next slide, please. And I also want to actually just make the point that we have to present the data to the clinician in such a form as they can understand it. Munir made the point that actually the average clinician has 10 minutes to see a patient. So if he's going to integrate multiple different sources of evidence, it needs to be presented in a way that he can quickly assess it and communicate the information to the patient. So that's an additional challenge. The meeting focused a lot on differentiating causality from coincidence. As you collect more and more data and more different data sets, there's a higher and higher chance that they will generate coincidental associations that have no causality underlying them. Next slide, please. And this is a nice sort of illustration of how conflicting results creates uncertainties and actually then confuses a patient. Next slide, please. And this is a nice illustration of some of the issues from an IMI project, PROTECT. And in this study, they looked at the sources of variability in multi-database study, which is really pertinent to today's conversations. And in this study, they looked at 6 drug ADR associations across several European databases. And here, I just show one of these associations, the association between antibiotics and the risk of acute liver injury. And the study did a joint development of a common protocol, and then they independently conducted it in different databases. And I think you can see here that while the relative risk is across the different study designs and databases were predominantly in the same direction, there was a significant variability in the magnitude of the effect estimate. And note that this is a log slide. And if we start to unpick this a little bit, next slide, please, you can see that if you look at study design, again, there was a consistent direction of the effect estimate, but a varying magnitude. So study design should be a really conscious decision when you're looking across different databases. Next slide, please. Here, looking at the outcome, the stringency and accuracy of definition increased the strength of the association. So less stringency led to more false positives. So outcome needs to be really carefully defined. Next slide, please. Here, the time window of exposure in the bottom left-hand side had a substantial impact on the relative risk. So really careful definition of the exposure window is essential. Next slide. And here, the study population, the disease stratification, comorbidities, medications, adherence, and the methodology used for matching. Next, please. Confounding adjustments. The authors found that the databases varied substantially in the lifestyle factors recorded, and that made the quality of their measurements really difficult, making comparisons between different databases difficult. Next, please. And finally, the databases themselves had an impact. I don't have a slide for that, but fundamentally the accuracy and completeness of data across different parameters is variable, so a systemic evaluation of strengths and limitations is essential. Next, please. So this brings us to the regulatory challenges. Next, please. And we are starting to move from a situation where we have structured data generated in accordance with strict guidelines and known provenance, so high certainty. We're very comfortable with that. But we're being asked to move towards, next, please, the use of more uncertain, unvalidated data of unknown provenance, and that generates more uncertainty in the regulatory decision-making. And these decisions are not hypothesis-generating, they're impacting directly on public health. So certainty is needed. Next slide, please. And I like this, because looking to the future, you know, this is going to increase, as I touched on earlier. This shows you a visualization of the topology of complex data from a consortium of adult severe asthma cohorts. So imagine this coming in as defining a patient population for study, and there would be numerous questions underpinning the use of that data. Next, please. You know, how are the individual components validated? How reproducible are the cohorts? We might ask, how is data weighted within the algorithms to generate the cohorts? And how stable is the measurements over time? And do these translate to really a defined patient population? Next, please. So our aim would be, and the meeting discussed about, how do we generate certainty for regulatory decision making? Next, please. And we have to be really careful how we present this data. This was a recent study that was received a lot of attention in Europe around dementia prevention, intervention and care. And the authors were careful in this. They looked at lots of studies and they developed a model, a predictive model, to assess which risk factors led to dementia. And they had a disclaimer that the PAF model assumes a causal association between a risk factor and dementia, and a causal link is required to lead to a decrease in dementia, and that RCTs would be required to give causality, but that would be very difficult for many of the risk factors they identified, such as education. But how is it picked up in the media? Next slide, please. And you can see that the media picks it up with 9 lifestyle changes can reduce dementia risk. So we have to be really careful about how we communicate the results of big data study, because it raises significant expectations. Other headlines were 30% of dementia cases could be prevented. So I think that this is it talks about risk communication and how we communicate the impact of these studies. Next slide, please. So what would be the solutions? I guess that's what you're all asking. Next slide, please. And really, we need to move, as I said, from a situation where we're using structured data towards considering the challenge of using this unstructured data, turning the data into knowledge with more certainty. And the solution here is really to develop a deep understanding of the data, to define the strengths and limitations so that the evidence arising from its analysis can be appropriately challenged. Next slide, please. And this is complex, it's difficult, it's not going to be easy. And there are multiple solutions and multiple directions. Next slide, please. So we need to think about the interoperability and harmonization of this data. We need common data models, and we'll hear a little bit about that today. We may need minimal data sets so that the same data is collected across different sources. And we need harmonization of standards. Next slide, please. We also need to really document the strengths and limitations of the individual data contributing to any insight that is derived, so that we get a robust validation, so we could be confident of making decisions based on this data. Next slide, please. But we also need to be able to access the data, and this is a real challenge. We need data for the common good. We need to be able to access and integrate the data, which is a huge challenge, especially with clinical healthcare data. Next slide, please. And that touches on or raises the issue of addressing privacy and governance upfront, and ensuring that data can be shared and that the public are happy to share their data. Next slide, please. So this brings me to a new regulatory initiative in the EU, which started in March of this year. Next slide, please, which is a HMA-EMA joint task force on big data. And as you can see, it's chaired by Thomas Senderovitz of the Danish Medicine Agency and has contribution from across the member states. Next slide, please. And its mandate is very broad. We started off by trying to characterize the relevant sources of big data, to define the main format in which they may exist, and we're asking lots of questions. What is the data structure? What is the terminology? How much of the data is structured? What is the standardization of the data? What may be its provenance? What's its geography? What's its quality? Quality means many things to different people. To understand the integrity of the data and its veracity, how heterogeneous is it? What is the variability? And what is the speed of change? How can we actually future-proof our regulatory system to deal with this? What is its rate of accumulation? We will be also asking completeness of the data and the opportunities to capture the data, how processed it is, that varies across different data sources, and what are the opportunities for linkage and combination, and what may be the opportunities for future exploitation. And also we need to ask about sustainability. With these multiple initiatives which are being generated, how sustainable are they? What's their refresh rate, and how can we ensure that the data is valid? We then will move on to identifying areas of usability and applicability across the product life cycle. And as part of this work, we recently launched 2 surveys to inform our thinking. We need to understand the constraints in the use of the data, to identify areas where we know additional data is required as a gap analysis, and also where the technology can really add additional value. Where do we need to drive targeted data collection? I think that's very relevant to the pediatric situation. And how can we drive validation approaches which will reassure the regulatory community of the robustness of the data? And we're then going to hopefully generate a list of recommendations and a big data roadmap. Next slide, please. So in conclusion, next slide, please. We have vast amounts of healthcare data that are continually being generated, and this offers us huge opportunities, but it makes it almost impossible to keep pace with the information. And we really need to harness the potential of the big data, which is generated by researchers and regulators, and that's hindered by the fact that it's unstructured, it's noisy, and it's inaccessible. Deciding which data to collect really starts by asking the right questions about the benefit source and the problems faced, and we shouldn't be collecting data for the sake of it. As I touched on, access to data is a really significant hurdle, especially for the observational data sets, which is the focus of our meeting today. But we need to look to the future also about how we can integrate in new data sets to generate that holistic picture and to generate further insights, and also to generate meaningful knowledge. But I want to finish by that validation of these associations is key to ensure the data can be used to support regulatory decision making. Thank you very much. I think, that's my last slide, I think.
Thank you, Dr. Kay, for that very exciting talk on an EMA perspective. And I think at this point we'll take a break. Why don't we come back at 10:15? I think we'll get started again. I'm not sure if everyone's in here. Okay. The next person who's going to be talking is Dr. Mark Hoffman from Mercy Children's Hospital. Dr. Hoffman serves as the Chief Research Information Officer for Children's Mercy and the Children's Research Institute. He has a lot of experience with Cerner, and we'll be talking about Cerner. And the title of his talk is How Can We Use Medical Records to Create Big Data for Analysis of Safety Signals? I just wanted to mention that Dr. Hoffman has to leave early and won't be able to join the panel at 1:50. I'd like to try to be able to ask questions of him after his talk, directly after his talk. Thank you.
And thank you for the introduction, and thank you for the invitation to speak today. I'll do my best to leave some time at the end for questions and answers. Many of my comments will be informed by my 16 years of experience at Cerner, but I am no longer there. So, but if you were to Google me, you'd see me listed on a number of Cerner patents. I don't see a penny from any of those, so there's not exactly a strong conflict there. I thought I'm going to focus on a variety of secondary uses of electronic health record data and use the work, some from infectious disease surveillance, some from research, to inform a perspective that I think will be useful for the goals of this conference. I've found that it's helpful to step back and think about the primary uses of EHR data, and that's to support point-of-care decisions during the delivery of healthcare. It's to enable immediate access to patient documentation, promote compliance, protect patient privacy. I actually see EHRs as playing a key role in protecting privacy, automating and streamlining clinical operations, and billing. It used to be that was considered the only use of EHRs, but really currently it's much more in a broader context. The perspective of many people who have worked in hospitals that use EHRs is that you put data in, but you never get the data out. How many of you in patient care settings feel that way? So I think we're starting to see the ability to actually do work with clinical data extracted from electronic health records, and that actually can help put the meaningful in meaningful use and start to generate value. So I'll describe a few scenarios. So some of the examples of secondary use of electronic health record data can be surveillance as an early warning system, investigate patterns of patient outcomes, inform quality improvement exercises, improve operational efficiencies, generate new discoveries, and ultimately the goal is to develop predictive models so that we can actually modify and catch risks before at the best time that's possible to intervene. I'm trained as a microbiologist, so many of my examples will come from a microbiology perspective. One of the earliest initiatives that I was involved in that demonstrated secondary use for surveillance was for public health surveillance. Many pathogens require reporting to public health agencies, and that process a number of years ago was a very paper-based, slow process. In 2001, when the anthrax-tainted letters were sent to media, members of Congress, we initiated a project in the Kansas City community to try to automate the communication between the diagnostic laboratories and the public health departments. And so we developed an architecture that extracted data from the laboratory systems, from the electronic health records, and packaged it and delivered it back to the Kansas City Public Health Department. And we found that that architecture is in many ways a very scalable concept. And we found some efficiencies that are worth thinking about as we consider drug safety surveillance. So the data completeness of this direct model was demonstrated to be considerably better than the standard process of faxing data or phone calls between the providers and the health departments. We also found the timeliness was much better. So when we thought about some of the bioterrorism scenarios, the average of 2.5 days between completing the result and that result reaching the health department was too long. By automating it, that transfer was reduced to less than a day. And so the ability to do near real-time surveillance is a really important feature of secondary data analysis. And then we also found that we reduced underreporting. There were comments earlier about fares and errors and self-reporting systems. So underreporting in general is a really significant issue that can be addressed. A few years later, during the 2009 influenza scare, we ramped it up to a national level. And then we worked with HHS and CDC to develop a system where we enrolled 850 sites around the US from 49 states to participate in an automated influenza surveillance initiative. Every site that participated received access to the geocoded maps. They received access to the trending graphs. So it was a really interesting model where in lieu of receiving a very light data packet sent into the system at Cerner, we provided value back to those contributors. And we found that that system informed a variety of other continuing data sharing initiatives. And through that system, we processed 57 million clinical cases during about the 6-month period in which that was a very active health concern. So I'm going to take a little bit of a detour, but I think it'll be helpful. So through working with public health organizations, there were some hard lessons learned where the expectation in a public health and epidemiology mindset is for a very clean and well-managed and maintained data set. So I've started using comparison of what I think of as gardens versus jungles. And so in public health, I think the appetite is really for a garden model. So the data is captured using instruments that are designed by experts. The weeds are pruned out. So you review the data, you evaluate data quality. The data is validated. So I have a lot of scars on my back around the expectation of taking EHR data and validating it and then delivering it to public health consumers. And that's just, as you'll see from some of my comments in a few moments, that's really not operationally feasible. The garden model is very labor intensive, so it is a lot of work to maintain a garden. And with few exceptions, they're relatively small in size. One great example is NHANES. So the CDC has an excellent resource that has been very successfully used for a variety of research initiatives in which they have 5,000 people enrolled in the NHANES initiative. And they're surveyed on average every year using a very well-maintained instrument. And it captures socioeconomic information, demographic information, healthcare information, some lab tests. So they can actually draw blood from the participants in NHANES to look at biomarkers and other things that may not be routinely captured during healthcare. If you're working with NHANES, you have the ability for any data element to look at a particular year and know whether or not that was included in the survey instrument for that year. So you have the opportunity in your data analysis to really go back and say, okay, I need to exclude allergies for 2003 because it wasn't part of the instrument. And so you have the ability to really control and manage your queries in a very rational way. The survey is very tightly structured. Every surveyor asks the same questions. The coding is consistent. The surveyors are trained in how to ask very formalized questions and generally gain the same response from the participants in the survey. And when we think about drug safety with registries and other vehicles for capturing data, there's quite a bit of similarity to a standard public health model. Some of the limitations, though, are the small sample size. It's difficult to scale because it's very labor intensive. So if you want to scale a survey-driven data capture process, you have to hire more people. And that's not always feasible. And then you could question whether there's discord between what people say versus what they do. And I think, so that's also an important concern. The jungle, as an alternative, is the actual healthcare system, warts and all. And so in healthcare delivery systems, despite the existence of recommendations and required processes, those aren't always followed, and that's where we have real-world data. There's limited standardization, limited data validation. There's wide variation locally and nationally among sites, and even within sites, there's considerable variation in practice. But there's far more coverage possible. And as we'll see, the opportunities are impressive, but there are hazards. So when you think about jungles, there are life-saving findings possible in a jungle, but there are also dangers. So the corner of the jungle that I have the most experience working with is a data set called Health Facts. A subset of Cerner clients voluntarily provide data rights to Cerner to use a de-identified portion of their electronic health record data for research. And this initiative began in 2000, so we have a pretty deep cohort. There are now about 860 healthcare sites that are contributing data into the Health Facts data set. These are everything from academic medical centers to private hospitals to physician practices, small rural hospitals, 15-bed sites, county hospitals. So the whole gambit of and variety of healthcare delivery is represented within this data. The sites are representative of roughly 100 organizations. So for example, an organization that has 10 hospitals will be counted as one organization with 10 sites. I'm not very familiar with it, but I think Epic has an emerging initiative called Cosmos. So if you're an Epic site, you may look into that. I'm not sure exactly where they are in the life cycle of that, but just to be as agnostic as possible, that's an alternative that's starting to develop. So as I noted earlier, Health Facts is a subset of the Cerner client base. The data is extracted every night through an operations job. It's de-identified, mapped at Cerner, and bundled into the Health Facts data set. The skill set needed to analyze the data is SAS, R, or SQL. There's not currently a user front end for working with this massive data set. Some statistics on the Health Facts data set, and this is where the magnitude of the opportunity starts to become clear. There are about 63 million unique patients represented in the Health Facts data set. The staggering laboratory data, 4.3 billion laboratory results. We have about 734 million medication orders represented in the data set, 489 distinct diagnoses, and a large number of other data elements represented within the data. Some other things that are more recently added: vitals, pain scales, falls, BMI, provider specialty, so you can compare nurse practitioners to physicians for certain metrics, APGARs. Meaningful use required the documentation of smoking status. So if you were analyzing the data, you'd see a huge surge in smokers, but it's really just because the documentation was increased through compliance with meaningful use. There's also a small subset of the subset that is providing surgical data. So about 20 sites have ASA scores and other really granular surgical metrics. So I'm going to share a few brief examples just to give a flavor for the possibilities. So Health Facts can be utilized for hypothesis-driven work. So this is an example in collaboration with St. Luke's Hospital in Kansas City, where their question was, magnesium supplementation is recommended after an AMI, but they identified that the evidence supporting that practice wasn't very clear. And it may be difficult for those of you in the room to see the graph, but the finding was that we had, after the inclusion and exclusion criteria, there were still 11,600 patients in the system that their data qualified. So when you think about doing a standard study versus this model, that's a very significant number of patients to work with. And the bottom line was that we found that both high and low magnesium levels correlate with a higher risk of in-hospital mortality. Now, that leads then to what is behind that. And so that's where you can do further research based on that. There has been some drug safety work done. So this is a paper in which adverse events and hospital length of stay associated with amphotericin was evaluated. I'm very active in a project that's CDC funded right now, where we identify a quality improvement project candidate at a collaborating site, Truman Medical Center in Kansas City, and then we compare Truman's status for that metric to all of the other sites in the HealthVax data and use that to prioritize our project. So if we're in the bottom quartile for a project, that raises the priority of that candidate. If we're in the top quartile, we may not put that as a priority. The real value of big data analysis is to do what I think of as letting the data speak. So instead of asking a specific question, you can use regression analysis to rank risk factors. So this example is a poster around roughly 20 factors associated with risk of hospital-acquired C. diff infections. And there were some factors in this data that correlated with lower risk and quite a few that correlated with higher risk. But instead of asking the specific question of what's the correlation of genitourinary infection, which is just one row here, by asking all of these things, that can guide, again, your future analysis plan. So in thinking about the role of EHR data for drug safety analysis, what are some of the things we need? One of the key strengths of electronic health record derived data is that every event is very precisely date and time stamped. So you can map the sequential order of ordering a medication and then the downstream effects. Hopefully it would be a favorable response, but let's say there was an adverse response. Some of the handles that we have are we can do a traditional diagnosis code analysis. So if you were only working with claims data, that's really the majority of what you would have to work with. So for example, you could query for patients with Stevens-Johnson syndrome. Where you have to be more creative is thinking through what are some proxies for adverse events. So one would be, was the patient prescribed Benadryl? And that's a possible proxy for an allergic reaction. Or if you, as we do in this case, have access to billions of laboratory results, you can look at liver panel profiles and evaluate, was there liver toxicity after the medication was prescribed. If you think about Vioxx, nobody was querying for cardiology-related adverse events. And when you do that, let the data speak model, that's where things that are unexpected can pop out. So you can see unanticipated trends. Retrospectively, some of my colleagues did an analysis in this data set, and they did see potential signals potentially indicating that that Vioxxing what could have been predictable. So the another category of adverse event that I'm very interested in is failure to benefit. So we don't really spend much time thinking about that, but as we're talking about pharmacogenomics or other indicators, many pharmacogenomic markers aren't that the patient will have a risk from the drug, it's just that they won't benefit from the drug. And increasingly we'll have handles to identify that. With timestamp data, you can also be more rigorous about excluding patients. So, for example, if you're looking at liver toxicity and you want to exclude patients with a prior lab test indicating hepatitis, you can go back over the longitudinal data and try to filter out patients who may have other indicators that mean that they should be excluded. So now to talk about some of the hazards. So what is missing from de-identified EHR data, especially in these large data sets? First of all, it takes if there's a new medication, the life cycle to get into one of these data sets is about 1 to 2 years. because of all the mapping processes that happen behind the scenes. Because this is a de-identified data set, no text notes are included because you can't reliably de-identify textual documents. So that's a major limitation. The data set that we work with does not have a large number of outpatient medication. That's also a very major limitation. It will be addressed in future releases, but currently that does constrain. And then compliance data. We don't have visibility to retail pharmacy and how often the patient's actually filling their scripts and so forth. Also did an analysis with John DeStasia at VCU, and we compared health facts with the HCUP NIH National Inpatient Survey. And overall, we found that there was a high correlation between the two data sets. The two areas that were different were mental and behavioral health and what you could categorize as women's health, maternity and other things. And if you think about the sensitivity of the information, you could probably back into why some sites may not be as eager to put those areas into their EHRs. So lastly, some of the other pitfalls. So this is a schematic showing if you were doing research and found this patient, you would be really excited because you found a patient that had Q fever, type 1 and type 2 diabetes, peripheral neuropathy, lymphosarcoma, and all of the other conditions that I showed here. The challenge is if you don't understand how EHRs are supported and maintained, you probably would have started to write a paper about this. But if you do understand how EHRs work and how they operate, when you're testing a new feature, you create a pool of test patients that are not real people. And you load them up with every crazy disease that you can. And it's often done by people with a technical, not a clinical background. That can sometimes get into your data set. So, and that is the case here where there are a patient category of test patients. So you have to filter out things like that. But you only know to do that if you understand the EHR nuances. So to conclude, there are major opportunities to do safety analysis and research with de-identified electronic health record data sets. But you do need to apply a team approach with subject matter experts who understand the workflow, who understand the clinical subject matter domain, but also technical experts who are able to navigate the data structures. And so really a team approach is critical to successfully doing this type of work. And these are some folks I just want to acknowledge. Thank you.
Thank you, Dr. Hoffman. I would like to open it up to questions for Dr. Hoffman. I wanted to just say that when people come to the microphone, if they could say their name and their affiliation, where they're from. I do have a question. How do you approach data cleaning in a system such as Health Facts?
Right, so that's a really important consideration because data cleaning involves understanding the nuances of the data. So with the example that I just shared, purging out test patients is not likely to have any adverse impact on an analysis. When you start to get beyond, and then you can also do things to handle missing values, or if you find somebody whose weight is listed as 1,000 kilograms, or things that are just completely infeasible, you can flag those entries and manage that, but through traditional data science methodologies. But you just have to really be careful and have a combined team that's both a data science perspective and clinical experts. So, there may be test patients with Ebola, and what you wouldn't want to happen be if there's an actual patient with Ebola, you wouldn't want to purge them out of the data, but you do have to really scrutinize those outliers. Looks like there's one more question.
Patrick Ryan, Johnson & Johnson. So first of all, I have to say I dig the fact that you did the screenshot of Pitfall. That was awesome. At least I appreciated that.
I had to flash through that, but yeah, it's a little throwback there.
Oftentimes in pharmacoepidemiology studies, we're worried about the continuity of follow-up in longitudinal settings. And oftentimes some people would advocate that one of the primary values of having something like an administrative claims system is the notion of an enrollment file, whereby observing something suggests the presence of it, but also not observing it in some way suggests the absence. So it's not missing data, it's actually informative that there's nothing going on. And that's often been a criticism laid at some of these EHR systems. Could you just comment on your take on that, and specifically strategies you guys or the community should be thinking towards to kind of get over that dogma?
Yeah, so the whole absence of evidence is an evidence of absence issue. And there's not a great way, when you're working with these data sets, that's where you really have to understand how the data got in, what to expect and what not to expect from the data. Compliance is a really difficult thing to infer from electronic health record data. So there are some things that are not ideally suited towards working with these data sets. And that's, as organizations and vendors move to more of a population health model, they are gaining increasing visibility to claims data and some of the other things that are not exclusively in the EHR domain.
Thank you. Ellen Werner from the National Heart, Lung, and Blood Institute at NIH. I'm particularly interested if you've had any experience with the newborn screen population where children or babies are diagnosed through newborn screening, then brought into care, such as at Children's Mercy, and then they're followed clinically, and how you can use those administrative data sets from state departments of health in combination with the EHR data. Do you have any lessons learned for us?
Yeah, I think that's something that's always been an aspiration of mine, is to get better access to newborn screening data. So that's really the one of my other areas of interest, is integrating genomic and EHR data. And but because typically the newborn screening is performed by a state lab and sent as a paper report rather than sent digitally, the opportunity to do this level of data analysis is still pretty limited. I would love to see more progress towards a digital submission of discrete findings from those state labs so that it's more seamless.
Thank you.
Yeah. All right, well, thank you very, or was there another? All right, thank you very much.
Thank you very much, Dr. Hoffman. Next is Dr. Adam Perrer from IBM. Dr. Perrer is a research scientist at IBM TJ Watson Research Center, where he's a member of the Healthcare Analytics Research Group. And he's going to be talking about data-driven healthcare, visual analytics for exploration and prediction of clinical data.
Thank you very much for having me. So yeah, as Ann said, I'm going to be talking about some visual analytics approaches for making sense out of big data. And sorry for the delay. I'm actually trying to use my own computer so I can maybe even give a live demo of some big data stuff that we're working on. But basically, I want to start off my talk by just kind of giving kind of an oversimplified cartoon, if you will, of kind of what is motivating a lot of our recent work. And essentially, you can imagine a scenario where you have a patient that's coming, exhibiting some symptoms to a clinician, and of course, the clinician has access to their experience, their expertise, their education to kind of come up with various hypotheses about how best to treat that patient. But of course, we're all here today because we want to leverage this big data to see if this can help kind of provide additional data points to this decision-making process. And so we have these EHRs that are filled of millions of patients, as you just heard is an example of one of those, where we have lots of features for every single patient, whether they're demographics or diagnoses or lab tests. And the idea that really motivates the group that I work in is really trying to build tools to help us search and analyze those patient databases to find patients that are relevant to us. But in addition to kind of coming up with these automated search approaches, we also want to build tools for the domain experts, the clinicians themselves, to kind of use their expertise via interaction to kind of further refine and filter those results, if you will, so that we can be ensured that the patients that we're actually doing analysis on represent who we're trying to find. And so once we have these kind of refined results, then we can then do some stratification and do some statistical analysis to figure out, okay, which treatments have worked retrospectively well for these patients that match the patient we're trying to treat, which ones do not, and maybe this can be an additional insight to help guide the decision-making process. So again here, the idea is not to replace clinicians, but really to kind of empower them with all this data that's being collected, as we've talked about today. And so my team at IBM Research has really built a lot of visual analytic tools to kind of help make sense of this. Given the limited time here, I can't talk about the whole suite of things that we've done, but basically the idea is, can we present this information in a visual way so that we can start making sense out of it? So really the idea here is how do we communicate what's kind of hidden in the big data so we can make sure that it's the analysis that we're doing on top of it can make sense. And so I'm going to just talk about in this brief time I have with you today, example of two tools that we've worked on that I think kind of somehow will be applicable to a lot of the things that we're discussing during this workshop. And so by no means is this the extent of what we do, but nonetheless we'll give you maybe a taste of how such types of analysis might be able to help you in the type of work that you're trying to do. So if we're thinking about electronic health records, really if we think of it as a very basic sense, imagine that we just have one patient in our data, right? And so this patient has different events over different periods of time. And so in this example, which is not a pediatric example, but imagine we have a patient with heart failure where they had antihypertensive drugs, and then a few weeks later maybe took beta blockers, and then a few months later took diuretics. And so if we can extract information like this from our EHRs, and we can start to do some analysis on it. But of course, once you start having many patients that take treatments in slightly different orders and they take slightly different treatments, even with just a few patients, it starts to become a little bit complicated to start finding the trends in the data. And of course, on top of that, we have different outcomes associated with each of these patients. Some of them might be well managed in relatively successful treatments. Others might have been hospitalized or had other negative outcomes. And so we need to be able to figure out, okay, what are the paths of treatment that work for the patients that did well, and what are the paths of treatments that didn't work for the patients that did not do well. And so in this particular case, I'm just going to show an example with heart failure. And the reason why I'm choosing it here today is it's a really common disease. It happens to a lot of us, and it's difficult to manage, so there's not really systematic guidelines for how to treat a generic heart failure patient. This is mostly due to the fact that the presence of all the additional comorbidities can really affect treatment recommendations. So this is one scenario of an example of how we can maybe leverage big data to make some progress in this. And so again, imagine we have hundreds of thousands or even millions of patients diagnosed with congestive heart failure. What we're doing is we're taking all this information from the EHR, so all these different types of features I was talking about, whether they're treatments or lab tests or diagnoses and so on. And what we're doing is we're kind of aggregating them in a way where we start with that complex patient that we really want to try to figure out. So that example of that patient in that cartoon. And then what we have is we have some analytics where we can basically try to find similar patients to them. So I'm not going to get into too many details about the similarity analytics, but imagine if we could find similar patients to those that we're trying to find that have similar types of medication, symptoms, diagnoses, and lab tests that are already there. Maybe we could then, look at what happened to them after their diagnosis, align them all by when they got their disease, and see what happened after, and can we learn anything from that? And so behind the scenes, what we're actually collecting, I know it's a little hard to perhaps see on the screen, but basically you could think of if we had a very simplistic scenario where every patient that had this disease ended up taking 3 treatments, and they all took them in slightly different orders. Then some might have taken treatment A first, and then B, and then C, and others might have taken C first, and then A, and then B. So this is kind of like the data that we could collect behind the scenes. So this is not how I propose to visualize it, but the idea here is that we can basically compute statistics on all the different paths and permutations of these patients. We can figure out how many patients went down each path, how long did it take them to go down each path, and what was their kind of average outcome, if we can compute that as well. And so the way I'll be just kind of visualizing it here today, and again, I apologize for might be a little difficult to read from where you're sitting on, but imagine we have all the patients kind of starting here. So this is when all the patients that are relevant to our patient got diagnosed with heart failure. And we can see some patients ended up taking diuretics first, and other patients maybe took cardiotonics first. And this is just an illustrative example. I'll show you the real data in just a second, but this is just so you can learn how to read it. You might notice that some of these paths are green and some of these paths are red, and essentially that's how that corresponds to the outcome. So the greener the path, the healthier these patients are doing according to our outcome. In this case, we're measuring hospitalization within 30 days, but we can see a clear difference between these 2 types of paths. But of course, the paths get a lot more complicated. They won't just usually start with one treatment, but they might have another treatment, follow-up treatment. And so again, the way these paths work is that some patients after diuretics took beta blockers and so on. Others took antiandrogens and they didn't do as well, so it's a little yellower than the green, and others might have followed up. So you can start building kind of maps like this of what happens to the patient. So with that said, I'll actually show you a very quick data demonstration of how this data works on real data. And again, I know it's really hard to see on this screen here, especially from where you're sitting, but maybe what you can maybe make sense out of it is that there's a bunch of patients starting here on the left-hand side, and there's a bunch of different paths emanating to the right. And again, this is for all the different treatment variations that these patients went down. So I'll just highlight one particular path here that maybe is visible to you there. But basically what we have here is these are patients that ended up taking antihypertensive drugs first after their diagnosis. And we can kind of see that lots of different pathways to the right emanate from that. Again, it's probably hard for you to see, but there's different thicknesses of these paths, and that corresponds to how many people went down this path. And if you had access to my screen or you were really close to the front of the room, you would see that there's one in particular thick path. So for patients that ended up taking 4 treatments, the thickest path on the screen is patients that took this one particular set of antihypertensives, followed by beta blockers, followed by diuretics, followed by anti-angiotensin agents. And so the healthcare institution that we were working with, this was kind of their default guideline for how to treat a generic heart failure patient. And we can kind of see here, if you dig down and you could look at the screen a little better, you would see that these patients aren't doing that well. They're only about 2/3 of them were able to stay out of the hospital within 30 days. So given that, it suggests that this kind of typical pathway is maybe not the best way to treat, especially if you start looking around the room and you can kind of see much more greener paths that have much more higher outcomes with less treatments. And so the idea here is if we present this information in this way, could we actually start to learn about maybe we could get these patients on the better paths faster and right away, and maybe actually achieve additional outcomes. And so you can kind of think of this as a way to start exploring how the patients actually move through these treatments in your data set. So I'll admit I went fast just for the purpose of time, but again, would be happy to answer questions or show you later on a better resolution. So the main lesson I wanted to show you by showing in this tool is that visualization tools like this can really provide an effective overview of your data. I mean, number one, it can help you verify if your data is behaving expectedly. That's a really important thing. But the idea too is it maybe can even lead to some hypothesis generation tests as well. If you start seeing patients going down different paths that you didn't expect, or different types of events that you didn't feel were important, it might lead you to generate some new hypotheses as well. The next tool I just wanted to briefly talk about is a tool called Coquito. And here, this is really defined with a different challenge, and I thought it was relevant given the abstracts of the rest of the speakers. And really, it's about how do you effectively define a cohort in big data, which is really important for if you want to do some retrospective statistical analysis on how patients have been treated, or you want to do some machine learning approaches to do some predictions. And really, to define cohorts, and especially whenever I work with a hospital or a healthcare institution, usually there's a situation where first the medical researchers define the requirements for how they think the cohort should be expressed. And then technologists will go write various database queries to try to mimic what the medical researchers suggested, and they deliver them back. And oftentimes, this is a very long iterative process because maybe there'll be too many patients returned or too few patients returned to do the study, or the features of those patients don't really make sense, and the process really restarts. And so it's truly not an overstatement to think that this process can really take months, many months, in fact. And so we were inspired to see if we could, build some way to basically explore these big data databases kind of almost in real time with our medical colleagues, so we can kind of get some feedback to kind of do some quick analysis of, is there enough data for the types of patients we want to study on, and are we sure we're getting the types of patients that we want? And so really it was about kind of creating a visualization to allow us to kind of add constraints to our patients to make sure we're getting the types of patients we want. In addition to just having constraints of patients that maybe had conditions A, B, and C, you might also be interested in patients that had A, followed by B within 30 days, followed by C within 30 days. So those kind of temporal constraints, being able to define those is typically really hard to do with database queries, but something we needed to support for lots of the types of analysis that we were doing. And so I'll just kind of show you an illustration of how this kind of works in practice, where basically this is a relatively small database, but I'm just using it for the demo here today of just 200,000 patients. And basically at the very beginning, I'm starting with all 200,000 patients. And you'll see as we start to use this tool, the result set on the right-hand side will get smaller. So you can kind of think of it as we're building kind of filters or funnels to kind of get to the patients we want out of this whole database. And again, it works with larger ones, but I'm starting with a smaller one just as a demo today. In addition to these two big bubbles that represent the size of the patients we have, we also have some kind of overview information about the gender breakdown and the age distribution and so on. And below here, we also have, we try to summarize what are the most common features for each of these patients. So we summarize kind of, there's a bunch of, if you can see the screen better, there's lots of these kind of rectangles, and they're sized by how often they occur. And you'll see these rectangles will change over time as we start to dig down to more specific patients we like. Basically, the idea here is it'll kind of give us a quick overview of who these patients are when you kind of aggregate them at a high level. And so what I'm going to do today is I'm going to search for patients that have a specific ICD code for diabetes, just to kind of do an illustration of how we can start filtering from this 200,000 to the patients that we might care about. So we're going to look for a specific type of diabetes patients. So in a few seconds, we'll get back all the results that we have with diabetes, and you can kind of see we go from 200,000 patients to about 26,000 patients. So already our cohort becomes much smaller just with even this one single constraint. And what we can then start doing is we can click on this cohort that we just selected and start seeing how all the different views change. So you'll see the gender breakdown and the age distribution will change shortly. And we can start analyzing kind of who these patients are and are they starting to represent what they want. And as I mentioned before, the kind of these blocks at the bottom, which are probably hard for you to read, again, represent all the different diagnoses and procedures and labs that they have, and you can start to kind of investigate, okay, what are the more common ones for the patients that had this diabetes diagnosis? And this type of interaction will become more useful as you start doing more constraints and you get more specific patients, but they might give you clues for additional constraints to add to the query itself, because if you notice particular things that happen a lot with this cohort that you're interested in. But I'm going to go on and since I know what I want to search for, and I'm going to search for patients that had diabetes and then sometime after that had proteinuria, which is a common side effect of diabetes, which can sometimes lead to renal failure. So we're going to add proteinuria to our query. And again, within a few seconds, we'll start to see the results of who matches that. And now we have already down to about 1,000 patients out of this, originally much larger data set. And we're going to see patients that had diabetes followed by proteinuria, and then I'm going to add also patients that had renal failure here as well. So again, we're starting to build this kind of temporal constraint of where they have to have certain events followed by followed events. And again, we're getting really small here where we only have now 100 patients, 150 patients or so that actually fit this condition. And oftentimes you don't want to stop with patients that actually just have the condition you're interested in. Maybe you also want to ensure that they were treated in some way so you can do your analysis. And so you can add further constraints. So for instance here, I'm going to add some additional constraints of hemodialysis to see if they were actually treated for renal failure. And just to illustrate the complexity of how these queries can actually get. Maybe you don't care if they just had hemodialysis. Maybe you're also interested in if they also or had peritoneal dialysis. So you can kind of create these branching structures where you can kind of start saying, okay, as long as they go down one of these paths that we build, they're going to qualify for a cohort. And so again, being able to kind of do some analysis and see how many patients match your hypotheses can do that. And so again, it's probably hard to see from where you're sitting, but now we're down to like 25 patients that actually match either of these conditions, so which is relatively small. But again, you can decide if this is enough to do your analysis or not. And of course, you can click on who those patients are and start exploring them in more detail. And again, all the views will update as well. And the other thing that you can also do, which I won't go into too much right now, is that you could really do side-by-side queries. So if you wanted to kind of do case control analysis, you could start to ensure that the patients that that you just defined above with that blue query, you can make a corresponding red query that is maybe the controls that aren't those patients, but also had a similar path of treatment, but didn't have diabetes to begin with. And you could start feeding this to your analysis as well. And so really the point I wanted to make here is that really providing kind of easy and interactive ways to query this big data is really important to kind of put the domain experts in control of the data so they can ensure that the data that we're doing analysis on really makes sense. And providing these kind of intermediate results as they're building the query really provide critical feedback to both how much is the database making sense, how is the data stored making sense for your analysis, but also might actually provide additional refinements for how you might want to adjust your cohort based upon what's being returned from the database. And this is particularly important when data can get very messy, as Dr. Hoffman mentioned just before. So I guess the main takeaways here is that, again, big data is really complex and messy, and I really believe that tools like this that give us a kind of an overview of the data can really fill a much needed gap. They can help verify our data quality. They can help generate novel insights for additional future analysis. And with that said, I'll leave that there for you today. So thank you all for listening.
Actually, we haven't had this opportunity because people haven't had as much time, but I'm thinking that maybe people might want to ask you some questions.
Heidrun Hildebrand, Pediatric Development, Bayer AG, Germany. I may have some very practical questions because it's impressive what you show there. But the question for me, or maybe it's a very European question, is first of all, what kind of data pools you can really tap in here? What is the basis for this? Is that something like Kaiser Permanent, a single hospital, or one insurance company you can tap in? And how can you get access to the data in the price of data privacy, data security?
Right, yeah, so those are really good questions. Yeah, so I didn't go into detail for that just because I was trying to just give an overview of kind of what's possible. But yeah, typically we partner with various hospitals and healthcare institutions, so multi-site institutions that are able to provide us the data, and we kind of do joint research together and write papers together on describing the analysis. But yeah, we don't, so we try to build our tools to kind of be independent from that as possible. But yeah, but again, we're working specifically with the healthcare providers to get access to the data. Yeah.
I think I missed where the source of the data that you were showing.
Yeah, I didn't mention that just because I didn't want that to distract from it. One of the sources of the data that I was showing here is a healthcare provider that we partnered with, who I can't mention by name, but they're a major provider in the Northeast, and so we worked with them together to do joint research, and a lot of how we decide on the technologies we want to build are really based upon our conversations with those healthcare institutions. What are they struggling to do with data, and we try to kind of build solutions to overcome them. Yeah.
And the other one was in terms of your questions. What kinds of healthcare questions are well suited to this kind of approach?
I mean, I think you could make the argument that, any type of question where you need to kind of, make sense of the data, having a view into the data, having some sort of higher level summaries of what's actually happening in the data can be very powerful. At the very start, again, just to verify your data is making clinical sense, but then also to kind of reinforce it. So I really think of visualization as kind of, a core part of the entire, data science pipeline, where it shouldn't just be at the end when you have your results and you want to communicate them, but you also want to do kind of more exploratory data analysis where you want to be able to kind of start probing for questions and ensure that the data that you're working on is making sense. Because I think if you don't analyze the data before you do your analysis, it's really gonna, it can have adverse effects to your analysis at the end. Yeah.
Bill Cooper from Vanderbilt. Really interesting way of thinking about, particularly visualizing as we prepare data. One of the things that we're thinking about over these couple of days is particularly as this work applies in children, and we think often about the developmental trajectory of children, and some of the work with drug exposure, drug safety happens in neonates up through teenagers. What's the ability then to sort of get some of the fine tuning around some of those age-specific things, including where we have to handle things like weight-based dosing?
Right. Yeah. I mean, fundamentally, if you have access to the data, then you could, and enough data, the data is rich enough being collected to do some analysis, then I think these tools are agnostic for, age. You can put in whatever constraints you want. But I guess the question is enough data being recorded that actually answers your questions is really ultimately what it comes down to, right? And it won't have, there's no magic solution to, figure things out if they're not there, but yeah.
Hey, Adam. This is Ketan Mani, Kaiser Permanente. So one thing I was curious to know about is how has your work, or this particular thing with the Northeast provider, has impacted in moving the needle, essentially, into health outcomes, or is it more on the exploratory end that they're exploring, or is it addressing their quality needs, or operationals, or workflow? So which of the things, maybe that's, if you shed some more light.
Yeah, that's a really good question. And yeah, I mean, right now I would say, it's more on the exploratory sense. So I don't have any great stories of how this is being deployed in the hospital right now and they're actually using it. We're starting to move forward with some things, but I don't have any great results to share. But really, yeah, you can kind of think of this as kind of very much, work in progress, figuring out what's possible, what can inspire solutions, and so on. Yeah, that's a great question.
Thanks. Appreciate it. Thank you. Dr. Rishi Kalamaswaran is an assistant professor at the Center for Biomedical Informatics and Division of Critical Care Medicine at the University of Tennessee Health Sciences Health Sciences Center. He is going to be talking about big data in analysis of streaming physiologic data, implications for healthcare.
Thank you. So not too long ago, there was a conference at one of the BD2K, or Big Data to Knowledge Centers of Excellence at Harvard, where they collectively talked about various forms of data collection and the implications and the questions that would arise over the next several years to come. And one of the ideas, or one of the topics that they centered around, was the question, how do we securely collect and analyze distinct data streams in real time to collect medical, or to guide medical decisions? And so my hope is that today I will be able to provide some answers about what sort of decisions we have as researchers and as clinicians and as epidemiologists going forward to tackle the problem of big data, specifically within the real-time domain. So here's a picture of a neonatal intensive care unit, and I'd like to talk about the NICU because that's where I started a lot of my work. This is somewhat of a dated picture, so what you're seeing may not exactly be the current NICU, of course, but one thing is for certain, what you're seeing on both sides of the wall are tons and tons of medical devices. And these medical devices, all of them are producing significant amount of data. For instance, you have the ventilators, the patient monitors at the bedside, the incubators, the IV probes. All of these things are producing significant amount of data. And of course, squished between all these devices, you don't see is a premature baby or the neonate. And of course, the nursing staff at the side to aggressively tackle any problems that were to arise at any point in time, of course, guided by these medical devices and the data that they produce. Here's a picture of a baby connected to some typical array of medical devices. You have on the top left, you have the ventilator, and then you have the warmer, you have the ECG monitor, you have the IV pump, you have the oxygen saturation monitor, and all of them are producing significant amount of data. From the ventilator, which ranges from a kilohertz of data, to the ECG monitor, which can also touch kilohertz of data. And all of them combined, when you add all of that up, you actually get close to 182 million readings per day per patient. But of course, in practice, what happens is only a handful are actually recorded every hour for every patient. So that's a significant amount of data that's lost on a continuous basis. Physiologic data is very interesting, and it's very unique in the sense that unlike other sensor data, physiologic data is typically, it has a high amount of variance. It has various signal strengths, and it's frequently non-stationary. So unlike your environmental sensors where you can predictably know how variant they would be, with these physiologic sensors, the patient is always moving, or the patient may be sweating, or the patient might just rip off the ECG lead or anything of that sort, and that produces noise in the signal, which are incredibly difficult to try to understand and try to clean, especially if you're trying to understand that from a real-time point of view. And changes occur over time as the infant grows and as the infant matures from a biological point of view as well as a physical point of view. You have significant changes. And for the neonate, new normals are established on a day-by-day basis as a neonate matures. And as I mentioned before, there's numerous artifacts, both endogenous, which means that there are some systems within the body that are contributing to artifact, and then also exogenous, where our nursing staff might come by the bedside to perform some intervention, which then causes artifacts in the signal. And finally, a lot of especially the bedside sensor data was never meant to be streamed. It was actually meant to be visualized at the bedside. So vendors have created these devices, put them at the bedside for the purpose of clinicians to visually look at the signals and make decisions. They were not meant to be streamed and analyzed on a continuous basis. So numerous challenges emerge for data scientists and researchers who actually want to capture that data and use it for research purposes. And one of the common items that we've had is for instance, when a nurse walks up and zooms in on a monitor, and you get a portion of the ECG. Well, when it's outputted from the monitor, the top portion of the ECGs and the bottom portion of the ECGs are not outputted. They're just cut off. So entirely meant for visualization purposes and not really meant for analysis. So the goal is to bring analytics to the bedside, and there are several considerations for that. And one of the main considerations is that, of course, we want to access the physiologic data and we want to analyze every bit of it. And the goal is to lead to a personalized and earlier intervention. But there's significant challenges to that. And some of them have to do with the fact that patients often come in with multiple conditions. So you're not really analyzing signals for binary classifications, but you're actually looking at multiple multi-class problems. You also have multiple streams of data, not just one. You have the ECG, the photoplethysmography, you have the arterial line, you have multiple signals that are continuously generated and are available for analysis. And within each of those signals, multiple behaviors are actually unique to certain conditions, like heart rate variability and all of that exists within each of these signals that if you looked at them in isolation, you may not be able to really exploit the full potential of that data. And in addition to all of that, you also have the technical challenges like real-time synchronous medical device data being mapped to asynchronous clinical data. So for instance, a nurse might be entering something into the EHR based on an event that she captured an hour before, but by the time it gets into the EHR, it's an hour later. And so what is the challenge is how do you map that, especially when you're looking retrospectively? So I'd like to talk about some technical things at the start and then get into some case studies later on. So from a technical point of view, we have all of these sensors. Each of these sensors are producing various signals. Those signals can be broken down into a hierarchy of events. And at the lowest level, you have these primitive events like up and down and increases and threshold breaches and all of that. But collectively, if you combine the primitive events together, you can actually compose these complex events. And those complex events might be sequences. A tachycardia followed by a bradycardia followed by a tachycardia. All of these things are sequences that might be meaningful when you apply retrospective analysis. From an online perspective, that also is increasingly more meaningful, especially if a patient is having an intervention at the bedside. Those sequences and the permutations of the sequences become more of an interest. Those complex events, especially when they're grouped together across multiple sensors, form these multidimensional events, and those are even more important, especially when you do artifact detection. You want to actually capture events across multiple streams. For instance, the ECG and the PPG might present you heartbeat data, and using either one or the other, you might be able to identify regions where there's noisy data. And when you combine the multidimensional events with physical events that are actually taking place at the bedside, a nursing intervention or an IV line is being placed in, or there's an extubation, all of these things can be helpful and important to the analysis of signals. But the problem and the challenge continues to be that we only have, at this point, we only have a very limited window and very limited sensing capabilities to capture the full depth of multidimensional events and physical events. But I believe that as we go forward, we will become increasingly more capable of capturing these. For example, a camera at the bedside to automatically identify interventions by nursing staff. Or a connected to the bedside to identify movement or motion. All of these things are increasingly possible, and I think that we will start to embark on them, especially as we start to realize the importance of capturing those events. And typically what we used to do before is we would use the standard database approaches that you take all the data and you put it into a database, you analyze that over various intervals, you extract some insight, and then you apply the insight again on a batch basis. But recently, the core engine that we have used over the last 10 years or so, especially at my previous location, was the IBM Infosphere Streams engine, which moved away from the batch processing method and moved more deeply into the real-time processing, where each event is processed as it arrives. So you have a pretty much analytics in flow rather than analytics at rest. And more recently, since that work, a lot more interest has emerged around real-time processing of events, both in the business community as well as in the research community. And one of the most popular softwares or the platforms to use now is this Apache Spark platform, which we're now starting to use for real-time capturing of sensor data, and also use as part of our machine learning pipelines. And the approach that Spark takes is very much similar to batching in the sense that you have data, you run a query, you get some result, but it applies this over multiple time dimensions. So it collectively, they identify this as a micro batching process, where batches can be triggered every 10 seconds or every 1 second, and so it brings it closer to real time. The advantage of this is that as the data grows, your analysis or your query does not have to change. It can remain the same, but now it's applied on increasingly larger data sets. The results is also growing and becoming more adaptive. So you start to now add your results over time, and without making any changes to your underlying platform, you're now getting data that arrives and has been analyzed based on recent data. So at the Le Bonheur Children's Hospital in Memphis, we've started deploying some of this infrastructure. We have what you see on the far left is the data capture component, where we're pulling data from the Draeger deltas, capturing that through True Process Vines, which is a proprietary software developed by True Process, and feeding that through to our internal engine, which is built on Spark and Hadoop, and analyzing that data and passing it through to various consumers, such as visualization front ends, or even machine learning pipelines that are attached to other Spark clusters. So now I'd like to talk about some of the applications of all of this this platform and architecture. So initially, we started off with looking at very immediate events. So one of the events that we looked at was a case study called the baby crashing scenario. So ideally, you want to identify neonates that have a fall in oxygen saturation, that have a fall in mean blood pressure, and then you want to compare that to their gestational age. And if at any point in time, if the gestational age is lower than or equal to mean blood pressure for more than 20 seconds, and the oxygen saturation value is less than 85%, you want to trigger an alert in real time as the data is being captured, and then providing that result to the clinicians for real-time decision making. So we employed a tool, again using IBM Infosphere Streams, to capture data from the bedside, to analyze this data, and then we provided an alert that was used by clinicians in a research capacity to identify patients that needed their attention the most. And this was deployed within a very small cohort, but found to be showing promise. We then took that approach and expanded it to include more clinical data, and built a system around identifying hypoglycemia, using patients, their age, their continuous glucose monitoring results, and identified patients who were at various risk ranges, and provided means for supporting hypoglycemic patients on a continuous basis. The more recent approach is with apnea prematurity, where we looked at neonates who were apneic but unidentified. These are babies that were admitted to the NICU but had not yet been sent for continuous sleep monitoring. So we looked at a cohort of patients that had these spells, in other words, this cardiorespiratory events that were triggered from a combination of changes in heart rate, changes in oxygen saturation, and changes in blood pressure, and also changes in respiratory rate. We looked at those patients, and then we also had another question, whether changes within these cardiorespiratory events tend to precede neonatal sepsis. Which was a question that arose, and we wanted to use, as Adam had mentioned, a visual analytic pipeline to try to help clinicians better answer these questions. So in this case, we had to build the real-time streaming infrastructure in the back end, and also the front end engaging the interactive interface to help clinicians answer these questions, whether there is even relevance or relatedness between the two. The neonatal sepsis part, we looked at nosocomial infection. Typically, when you have nosocomial infection, you usually our neonates are given a cocktail of antibiotics, and they're administered even before diagnosis, just as precautionary measures. So ideally, if there's a way to identify patients that are at risk for nosocomial infection, then maybe you might not need to administer the antibiotics as heavily as one might do. And one of the key aspects of that is that there's been some research to show heart rate variability might actually be useful in identifying sepsis before it actually starts to show clinical signs. So we used heart rate variability as a component in identifying and generated that on a continuous basis. And then also we compared it to our neonatal spells algorithm, where we were combining various data streams to identify apnea and spells, and we combine them together into this environment that I'll talk about just in a bit. But before then, the algorithm for our apnea detection consisted of looking at continuous ECG waveforms, looking at impedance respiratory waveforms, and then looking at oxygen saturation on a continuous basis. And if there was a fall in any one of the three, we identified the sequences of the fall or a rise, and then we classified them into one of several types of apnea. And here's a diagram that shows we had 7 different classes of apneas that we classified based on sequences. For instance, at the very top, the central apnea would be identified as a central apnea if there was a fall in respiratory rate, followed by a fall in heart rate, followed by a fall in oxygen saturation, and then a recovery in respiration rate, and then a recovery in heart rate. If that sequence was identified, we would label that as central apnea. So we took the classifications that we made using the 3 waveforms to classify apnea, and then we took the heart rate variability and we combined them into a unified interface that was provided to clinical researchers to identify apneic events. And here you can see a PhysioX, which is a visual analytic interface for searching over the data that we had generated. In this interface, at the top left, you're seeing changes in breathing patterns. So the more clustered regions or the more dense regions are where more breathing tends to take place, and the y-axis is ranges from a breathing pause of 1 second all the way up to 70 seconds plus. So you can see in that diagram that there's significant breathing pause just under the 5-second area, but at certain times, the resolution is not great over here, but at certain times you can actually see that breathing pauses, especially in the top left region of the space, you can see that there's increasing breaches of breathing pauses, where many breathing pauses are observed in 30, 40, 50, and 60 second duration. There's also a red cross that's labeled just around the Tuesday 27th, 12 p.m. Time, just before Wednesday the 28th, right in the center. And that region was when there was a blood draw that was marked. That we extracted that from the EHR. We marked that on the visualization so that clinicians looking at the screen would know, well, what triggered, or would hypothesize what triggered the blood draw. And just before, if you look just where that cross is, just before that time, on the second chart at the left, the heart rate flux graph, which shows changes in heart rate variability, you start to see that just around that X region, there's increased heart rate variability and significant density between the 2 to 30% range. So in this case, the y-axis is a percentage. And just in the bottom, the oxygen flux graph, which are showing variabilities in the oxygen saturation, again, you see somewhat of an increased and decreased density around the 12 p.m. time, which indicates some fluctuations in oxygen saturation. In the center, you're seeing the actual waveforms that are displayed. So this is something that's familiar to the clinician. And the waveforms are activated when one of the classification icons on the right side of the screen are activated. When they're pressed, you start to see immediately which regions are causing, or what the underlying physiology is that is causing a certain event to appear. And zooming in, you're actually able to identify the sequences. For instance, in this image, you can see first the heart rate in the center falls, and then second oxygen saturation falls, and then you see in the third that there's actually some changes in respiratory, in the respiratory waveform. So increasingly, we're starting to expose the underlying physiology and making it easier to identify the machine learning classifications with physiology that's captured at the bedside. So a lot of the work I talked about before was based on rules and sequences. Now I want to talk about some of the work that we're doing that's mainly data-driven or driven by machine learning. Some of the approaches that we've started to work on is actually capturing changes in waveform on a continuous basis, so that can be applied within the real-time domain. And one of our approaches, what we call the probabilistic symbolic pattern recognition, the PSPR approach, where we're showing changes in amplitude and annotating them in real time based on changes in amplitude. So for instance, over here in this image, you're seeing that anything under 0.10 is classified as an A, between 0.1 and 0.2 as a B, and so forth. So when we capture all these symbols, we're now able to create a probabilistic matrix of the transitions between A to B, or the frequency of transitions between A to B, and also look at it from a collection. So what we frequently do is we look at combinations of several symbols and their transitions. So we look at 6 symbols transitioning to the 7th symbol, and what does that mean? And we found that when we did that, we were actually very successful in identifying proximal atrial fibrillation, and we are able to identify the normal patients who have no proximal atrial fibrillations to the ones that do. And we also showed that, interestingly, with our approach, sample frequency has a direct impact on how well the algorithm does. Interestingly, the algorithm tends to do better on lower sample frequency, which seems to indicate that there is increasing presence or increasing influence of noise, an artifact in the waveform that gets canceled out, especially as the smoothing takes place on the downsampling. And more recently, we've started also deploying deep learning methods, where in the previous instance we applied a feature engineering approach where we tried to derive features ourselves, but we also applied methods where we wanted to generate features through deep learning. So have the deep learning engine, especially the convolutional neural net, generate its own features based on raw data without us having to really influence it in any way. So the convolutional neural net is a type of neural net that has been designed especially for image processing or image recognition. So we wanted to apply that for ECGs, for waveforms. the typical approach is that you have an image and then you have convolutions that take place on each pixel of the image in accordance to its neighbors, and that convolution is then slided throughout the image on a continuous basis, and new features are extracted. So what you're seeing on the lower left is multiple feature maps that are generated from a certain image after the convolutional neural net has applied multiple filters. So one of the feature maps, for instance, might be identifying an edge. And as you go higher up in the hierarchy, these convolutional neural nets might actually be identifying increasingly more complex things like shapes and eyes and mouth and so on, and then classifying them all together at the end as a cat or a dog or whatnot. And this has been shown to be very successful in image recognition. So we applied the same philosophy for analyzing waveforms, especially the ECG. So ECGs are continuously sampled, and the prominent feature of the ECG here is the R peak, which is very evident, and you can, and it's continuous, so that these complexes are repeating over and over and over again. You can divide those complexes down to their individual complex, and that's shown on the bottom right over here, where you see the R peak, and then you see that QRS complex. What we did is we took a complex. Initially, we took a complex, and we took the signal, we normalized the signal, and we fed it through to a convolutional neural net. So what happens is when you take a peak and you chunk it up, you find the complexes and you chunk it up into individual peaks and you append them, you start to get these weird anomalies, these changes that just are way too far from the mean. For instance, you see this blue ECG that is just completely out of order with the rest of them. For most of them, you'll see that the peaks are generally identifiable. You can see that there's different levels of amplitude based on different time sets, and of course the influence of noise is very evident with these fluctuations that you see on the top and the bottom. And what we found is that when we applied a fairly deep convolutional neural net, where each of these, so let me start with the top left. The top left is your raw ECG that is about a batch of 60 seconds, and it's a one-dimensional image. So it's been flattened to a one-dimensional matrix, where each data point represents one of the samples. And over time, we're feeding that through to various convolutional layers, each of the layers here represented as a feature map. But you can see that the feature map itself, after it generates features on the previous data set, it actually does we do some very interesting linear regularizations, Batch normalizations, and then downsampling, and also do some dropouts to avoid overfitting. Over time, we feed the output of one of the layers into the input of the other, and so on through multiple layers. At the end, we classify them in the classification layer into one of four classes: the normal, the atrial fibrillation, other rhythms, and then noise. And we had about a total parameters of about 2.0 million parameters were generated by this deep neural net and used for the classification of the atrial fibrillation. So this was used for a PhysioNet competition where we had 8,500 patients, and they were all labeled according to one of four classes. And then we had a hidden set where we had to test this data on. And when we ran through the data based on our initial training set that was provided, we see that this method tends to do fairly well. So in the training set, we had 89.15% accuracy, and on the test set, we had about 84.69%. There's definitely some level of overfitting, which we are which needs to be further tweaked. But this score was able to achieve 3rd place overall in the unofficial ranking of the PhasioNet Challenge, just about 1.4% away from 1st place. So it's and when you think about it from a feature engineering perspective, we also applied 50 different nonlinear, linear frequency and temporal domain methods. And the maximum accuracy we were able to get on this data set was 0.80, or the F-score we were able to get was 0.80. And to get an F-score of 0.84 using automatic feature generation techniques is impressive. And I think there's lots of really interesting work that can be done in this area. Definitely, helping the convolutional neural net with pre-processing of the data, identifying the correct pre-processing levels are all potentially areas where we can further improve on our accuracy. So in closing, some of the take-home messages: is every breath, every beat important in the context of critical care. Numerous approaches exist for identifying event streams and their processing, but I think this is an ongoing and evolving area, and we'll have multiple tools that emerge. But in all of that, the most critical challenge here is collection of high quality data, things without artifacts. But again, as I had mentioned, I think this is an ongoing area of interest, and potentially more sensors or more data collection units in the ICU might actually help us overcome this problem, because right now we're only getting a horse's vision of what is going on in the ICU. Potentially more devices and even a video camera that constantly looks at movement and intervention and automatically associates that to events within the physiologic data might be something of interest. And the second point is that it's just not simply enough to analyze the data. We really have to pull in domain experts, and the ways that you pull in domain experts is not through showing confusion matrix and your loss functions, but through a more interactive visual approach where they are able to identify what exactly is going on. The other importance of a visual analytics approach, I feel, is that because as we start to use these deep neural nets and these black box methods, it becomes increasingly important to help our clinical partners see what triggered the alert and to try to make sense of that. It's extremely difficult to do that within the current deep learning methods that we have, and so maybe a tool like that might help. It also gives the data scientists a feedback loop about how they can further improve the algorithm and further improve learning. And finally, I'd like to end off with a note that there's been a lot of hype about deep learning. There's been a lot of hype about neural nets for several years, and I think that with some of the work that we're doing, we're starting to see real utility in the method, of course, with significant amount of hyperparameter tunings and all of that. But definitely, I think this is something that remains to be of interest for our group going forward. Thank you so much.
Thanks, Dr. Kalmaswaran. I just wanted to open it up. We do have some time before lunch if people have questions.
I wanted to ask if it's okay to address some of the presentations before the break? Or would you rather, yeah, would you rather we wait for those?
Maybe we could just address this one.
Okay.
Is that all right?
Sure.
Hi, Alison KVMA. Fascinating talk, really fascinating talk. I was particularly interested in the neonatal care data, which is an incredibly rich source of data, but also incredibly potentially confounded. And you focus mostly on the sort of individual patient, integrating the various streams of that. But sort of from a regulatory perspective, we sort of be thinking about the population level data. And so, you know, even the mind sort of boggles about thinking about the confounders within a patient. But what's your vision in terms of potentially adding and looking at it on a population level basis?
Yeah, I think that's a great point. A lot of our work has been driven by the idea that each individual is unique in the sense that they have a baseline, and then there's a change from baseline, which seems to trigger some clinically relevant event, and that a lot of our work has been to try to capture or predict the change in baseline before it occurs, based on subtle changes in the signal. But given that we've done all of that, there's obviously some common trends and some data that or some knowledge that we can extract and apply on a population level. We haven't been able to do that much because we don't have the data that supports this work. For instance, it took us almost 3.5 years to actually build a system that would be stable enough to continuously collect data from the patient monitors that we had in the NICU. And even after I went to the PICU, it took us, and we're still having difficulties, and it's been almost 1.5 years of capturing data. So it's extremely difficult, and I think that it's a great opportunity also, in that there are these open data sources like Mimic and all of these other things. But if there was an initiative that merged all of them together, then I think that would be very possible, and that'd be feasible as well.
And a sort of a follow-up question, I was also interested in your alert tools. I wondered how you built clinician trust in that, so that they would trust your alert to go to one baby and not the other. Yeah.
So I think that one caveat here is that all of these tools were deployed in the research environment. So these were mainly led by research clinicians that partnered with us and were also in the unit. So they would see all of the alerts, and they would, we would keep track of the accuracies and all of that. They were not deployed in the actual clinical domain. Having said that, one of our colleagues has actually done that. And it helps because he's the head of the PICU, so it helps to be in a position of power where you can encourage the adoption. But that's been fairly successful in reducing mortality of sepsis in the PICU at Le Bonheur. Trust is an incredibly difficult thing to gain, and it's a very easy thing to lose. So we always try to avoid going in somewhere where we absolutely will not be able to guarantee that we will live up to that level. So we are very much in the research focus. We are trying to build confidence through our research efforts, and I think that gradually over time we will get there.
I can't resist a comment before my question. In the 1970s, in the early days of databases, we just took the clinician notes and typed them into what really was just a word processor at the time. And the clinicians didn't believe what the computer was saying about their notes, which were actually verbatim what they had written. So, I think you're wise to think through the trust issue very carefully. Yeah. But my question was actually about that. In a time when the computational capabilities are moving so fast, and with tools like machine learning, which are not intuitive, where do you see validation and building trust even among knowledgeable researchers going? In other words, it seems to me it would be pretty easy to come up with a prediction that would be totally wrong and have no one able to do the math except the people who did the original experiment.
Right. And I think I totally agree with that point. And I would also add that what we find in our own work is the challenge is not really in the learning side and in the training side. It actually is in the data gathering and the data cleaning and the data validation side. So validation of data sets, especially when you train on and test on that data set, is so important. And that's why more and more emphasis has been on reproducibility of research results. That there have been instances where you may collect the best data and test on the best data, even if it's in these like competitions where you have hidden data sets, you might optimize for the hidden data set. So it becomes increasingly difficult because you're so narrowly focused on one criteria and optimizing for that one criteria. So it remains to be seen how these efforts will actually translate into the real world, where you are dealing with patients that might rip the leads off, or patients that might just walk off, where you're dealing with nursing interventions that might be unpredictable. So it's a very hard challenge, and I think that more and more in the community we're starting to appreciate it more and more as important, as essential to gaining the trust factor. But I still believe that the validation focus for any sort of machine learning in medicine should be on the data, and what data was used in the training and what data was used in the testing of the algorithm. Because that can really tell a lot about how translatable it is in the real world.
Okay, great. Thank you very much. And I think we'll take lunch now. And how about if everyone comes back at 10:01? Hello, I think we'll get started with the afternoon. The next person to speak is Dr. Patrick Ryan. He is a Senior Director of Epidemiology and the head of Epidemiology Analytics at Janssen Research and Development, where he's leading efforts to develop and apply analysis methods to better understand the real-world effects of medical products. He's going to be talking to us about large-scale analysis to transform the evidence generation process, lessons from the observational health data sciences and informatics or Odyssey collaborative.
Thank you, Anne, and the organizing committee for inviting me to this talk. And thanks for those of you who came back after lunch, even though I'm giving the next talk. Last year, I was told to come give a talk, and I totally broke the rules and just talked about my daughter. And I was asked to give an update. So my daughter's doing great. So that's very good. But what I have spent the year doing is reflecting on not just how do we do the mechanics of preparing observational data to conduct analysis, but how do we actually get information that's reliable and useful in clinical practice? And what I'd like to do is first introduce you to Odyssey for those of you who aren't familiar, but then walk through a few examples of very recent research that's been published, really as a challenge and a call to action for the community about where I think we might need to go as a group. So Odyssey is the Observational Health Data Sciences Informatics program. This is an open science community. Anybody is welcome and encouraged to join us. Our goal is to try to establish scientific best practices for how to use observational data to generate reliable evidence. The coordinating center is at Columbia University, but as you'll see, we've got researchers around the world who are working together towards this goal. Our mission within Odyssey is to try to improve health by empowering a community to collaboratively generate the evidence that promotes better health decisions and better care. And it's actually this specific focus on using data not as the end game of data standards, but actually just as the means to get us to the point of where we need to be that's really been the centerpiece of what we've been doing within the Odyssey community. Odyssey is an international community, and we actually have active representation around the world with particular, we've got a lot of folks here in the US. We also have a very large contingent in Europe, as well as in the Asia Pacific region. By last count, we have over 200 researchers from academia, industry, government, all engaged in 20 different countries. One of the important things to recognize as we bring up this discussion of big data is that we actually really demand a multidisciplinary perspective on a lot of these problems. And so we're excited by the fact that we bring together epidemiologists and statisticians and medical informaticists and computer sciences and clinicians together, because what we realize is none of them have the answers to a lot of the problems that we're wrestling with right now. Odyssey has adopted an open community standard, which is the OMOP Common Data Model, and to date there are over 60 databases covering 660 million patient records that are using this open data standard, and anybody else with data who would like to use that is welcome to. Even though we're talking about data and 60 databases and 660 million patient records and terabytes of data sounds like a lot of data, actually I think what's most important when we think about big data is actually having a big community. In Odyssey, we've been holding regular meetings, primarily based here in DC. Our next one's next month at the Odyssey community. But actually, as this community has grown and the excitement about big data has happened, we've had things pop up like a couple of us got a random invitation to go to Korea, only to find that an entire academic building was plastered with the Odyssey logo and hundreds of people were waiting inside. Or last month, there was a little Odyssey China get-together. But of course, nothing happens in China that's little. There's over 200 people who came to an Odyssey China meeting, specifically focused on how do we solve these big data problems. And what really came clear is this needs to be a community effort. Next year, there's actually going to be the first Odyssey Europe symposium. So if you are dying to join the Odyssey community but would like a better trip than to Bethesda, I encourage you to come to any of these other meetings. Otherwise, join us in a couple weeks. So one thing that Allison briefly touched on in her discussion is the use of a common data model. And I suspect that Jeff is going to talk a bit about the Sentinel data model in the next talk. Briefly, what the idea here that we've used in the Odyssey community and other communities have done as well is that we want to standardize the structure of data to be able to enable standardized analytics. Specifically within the Odyssey community, we've been trying to standardize the clinical data that might be necessary for pediatric research questions, but there are also other members of the community interested in questions around health economics, around health systems. And because we are an international community, not only do we have to standardize the structure of the data, which are represented here by these boxes, but we also have to standardize the content of the data. And for that, we impose a standardized vocabulary that lets us talk the same language, whether we're talking about diagnoses or drugs or procedures. So the basic idea is that this common data model can be applied to any particular source database, whether that source database be an electronic health record, such as the Department of Veterans Affairs, or PedsNet, which Rob mentioned earlier, or CPRD in the UK. If your source data was administrative claims data, whether it be from some of the vendors or from a large payer organization, or whether you're dealing with clinical data, such as the Ehr data or the NHANES that was mentioned by Mark earlier today. I see Josh in the audience. We also talking about clinical areas in the realm of things like the All of Us initiative that's going on. No matter what the type of observational data is, the idea is that a process is performed to standardize that data into a common data model. And what that enables is that there can be development of standardized analytics that are then sent to the database. Aggregate summary statistics are generated, and those aggregate summary statistics can be compiled into an open evidence repository. And this is the model that several of the efforts in Europe, like what Miriam has led in Europe and what Jeff's going to be talking about with Sentinel, and so too that's what we're doing here in Odyssey. Now, the approach that we're taking within Odyssey is first, we have strategy to focus on 3 different areas that we think are necessary to improve evidence. One is methodologic research. We need to establish scientific best practices and generate the empirical evidence necessary to believe in those best practices. And I would say in pediatrics, this is an area where we first need to figure out practices, then we need to evaluate to figure out if they're best practices. Within the Odyssey community, we're also trying to develop open source analytics. So it's one thing to establish a best practice. It's another thing to codify it in such a way that everyone in the community can consistently and reproducibly apply that best practice. And so we're developing software to do that. And then last but most important is we're trying to apply those tools and those best practices to clinical questions that actually matter to patients. Specifically within the Odyssey community, we've been focused on 3 different analytic use cases. The first is clinical characterization, so answering a descriptive question of what happened to them. So which treatment did patients choose after a diagnosis? Which patients choose which treatments? Think about this as disease natural history or treatment utilization patterns. And the key here is that characterizing a population means you first need to define your population. And so while we are here talking about pediatric research, in fact, I'd say that this capability is the exact same capability we would want no matter what your population is. We also want to be able to focus on patient-level prediction. So drawing inferences, not to just say what has happened to others, but what will happen to me. And Rishi provided a nice example of deep neural nets as an example of how machine learning techniques can be applied to data to fit a model. And then last but not least, we've also been focusing on population-level effect estimation. This could be safety surveillance, so does a treatment cause an outcome? Or this could be comparative effectiveness research. What we've recognized within the community is that these three problems are fundamentally different analytic problems that probably need different best practices to be established. And so what I'd like to do for the rest of my talk is actually walk through a few examples of recent research that I've seen out there that's really raised questions in my mind about, are we thinking about big data in the right way? And to do that, since we're here in DC, I figured I'd channel one of your own. So many people remember this quote from Donald Rumsfeld when he was giving a speech at NATO in 2002. He talked about that the message is that there are no knowns. There are things we know that we know. There are known unknowns. That is to say, there are things we know that we don't know. There are also unknown unknowns. Now he goes on into this to talk about this. Sounds like a riddle, but this is very serious, important matter. And the last paragraph usually gets dropped off when people grab this off of Google, but I figured I'd read it. Another way to phrase that is that the absence of evidence is not the evidence of absence. It's basically saying the same thing in a different way. Simply because you do not have evidence that something exists does not mean that you have evidence that it doesn't exist. And I want to challenge us as we're thinking about the use of big data, how much we're actually falling into some of these traps that Mr. Rumsfeld left for us a while. So to do that, I want to walk through each of these real quick. First, things that we know that we know. Just show of hands, how many people here think that we know that ACE inhibitors cause angioedema? Only like 5 hands do we think, wow. Or is everybody else just chilling on their lunch or? Okay, well, I think that ACE inhibitors cause angioedema because it seems like in every pharmacoepidemiology conference, people hold this up as the canonical example of a known safety issue that we have to prove that we can solve for. And maybe it'll even be in Jeff's slide deck too. The question is, let me make an assertion then, since not everybody's raising their hand. I will assert that I think we do know that ACE inhibitors cause angioedema, but what we want to know is clinical characterization. I want to know how often does angioedema happen in patients who are exposed to ACE inhibitors. Population level effect estimation, so what is the magnitude of that effect size? Or comparative effectiveness, how does ACE inhibitors compare to other antihypertensives? And then the third, which I'd say is actually the most important one that we actually do the least of, is patient level prediction. I want to know if I'm the one about to take lisinopril, what's the chance that I'm going to have angioedema? That's what we want to know. And I'm going to assert that this is one of the things that we think we know best, and yet we're kind of a far away from providing the evidence we need. So I'll just focus on clinical characterization, so it should be the easiest. So here is the product label for Zestrel, which is the branded name for lisinopril, which is one of the ACE inhibitors. And in the warnings and precautions section, it clearly states that angioedema of the face extremities, lips, tongue, larynx, including some fatal reactions, have occurred in patients treated with ACE inhibitors, including Zestrel, at any time during treatment. What you won't find in this paragraph is how often it happens. You won't know if this is happening 1 in 10, in which case maybe you don't want to take the drug, 1 in 100, 1 in 1,000, 1 in a million. This does not provide you that information. So I was kind of unsatisfied with that, so I went and found a generic manufacturer's version of the same product label for lisinopril. And in that label, further down in the label, it said, angioedema has been reported in patients receiving lisinopril. And then in parentheses, it says 0.1%. Now, I don't actually know if that is an incidence rate or an incidence proportion. I don't know what the time is, but at least I got a number when that's may or may not be better than nothing. So the product label isn't going to answer my question about characterization. So I went to the published evidence. And I found three studies, which you'll see why in just a second. The first study here, let's see, can I move that? Yeah, there we go. That's not going to go over there. All right, that top study there, it's a paper published by Miller et al. that took an observational data set from the Department of Veterans Affairs, and they found that the incidence of angioedema was about 2 per 1,000 person years. That second analysis there you see is McCanney et al. That's a meta-analysis of all the randomized clinical trials for ACE inhibitors, and it got an incidence rate of about 3 per 1,000 person years. And then the bottom one there is a paper by Darren Toe and colleagues from the Sentinel Initiative, where they used observational data from the Sentinel network, and they got an incidence rate of about 4.4 per 1,000 person years. Now, and that dashed line I put there just as a reference, that's 0.1%, if we can assume that that was on the same scale from the product label. Now, what you notice here is that all three of these things have very, very tight confidence intervals. And this is actually where big data can actually fool us quite a lot. You see, each of these studies had hundreds of thousands of patient-exposed years. And as a result, that gives us this false precision about the fact that there's very little sampling error in these estimates. However, what I see is the label told me 0.1, paper number 1 tells me 0.2, paper number 3 tells me 0.3, paper number 4 tells me 0.4. I'd hate to get another paper because the rates just keep going up and up. Now, what's going on here? Well, incidence rates are predicated on two basic assumptions that I think all too often we overlook. The first is that the observed data represents a random sample of a target population. And so if we're going to use data to represent, we're going to apply sampling statistics, we have to assume that this is a sample. Now, in the case of a Department of Veterans Affairs population, I'm not sure what that sample is. And if you would say, well, it's just the VA, so that is the VA, I'd say, why are we doing sampling statistics if you have a full population? Now, the second study there is a meta-analysis, so maybe that's okay, so long as you believe that the inclusion criteria are generalizable to the real-world population. Unfortunately, Allison told us that that might not be true. And that third study there is the one by Sentinel, and that is privately insured populations. All of these represent a population, but I'm not sure it's the target population. The second assumption that's really important here is that sampling statistics are based on the idea that an estimator is unbiased, so there's no systematic error. But of course, we're using observational data, and we heard from Rishi and from several of the other presenters, Mark, that these data are very messy. And messiness means that we can't actually be 100% sure that we're seeing all the exposures and we're seeing all the outcomes. And yet we make the assumption that it's perfect. So I would actually suggest that these numbers aren't necessarily bad. Maybe it's just the way that we're analyzing the data has a problem. So one more point of reference. How does this information get distilled to clinicians? So I pulled out of UpToDate, which is a clinician reference, what they talk about for ACE inhibitors angioedema. And they specifically say that the overall incidence of angioedema related to ACE inhibitors has been estimated between 0.1% and 0.7%. And they provide a whole bunch of references. So just to draw that, what that would look like, basically UpToDate at the top there is basically suggesting we don't really know. It's somewhere between 0.1 and 0.7. Now, what's fascinating is that's not a confidence interval. That is not a statistical artifact. That is simply a range that a series of clinicians decided sounded good to tell other clinicians. However, I actually feel like it's a lot more trustworthy piece of information than those summary statistics that we calculated with fine precision in each of our studies. In fact, what's interesting is this UpToDate thing, they mentioned 6 papers, but in fact, 2 of the papers were case series, so I don't know how that helps an incidence estimate. One of them was a clinical review that says angioedema is bad, and then 3 of them were these very 3 papers. So they generated the range of 0.1 to 0.7 on the basis of these 3 estimates that were 2, 3, and 4. I have no idea how they got 0.1 and 0.7, and yet I really like their answer to the question a lot better than what we're actually publishing. And that just makes me reflect on what we're doing and are we being fooled by the statistics we calculate from big data. So just to dig into this a little bit further, using the Sentinel paper as an example, I just want to visualize what this means so that you have a context for this. So in the paper, they show in the abstract that the incidence rate was 4.38 with a confidence interval between 4.24 and 4.54. And so what I'm showing you in this blue is a probability density function. That's just the estimate, exactly as reported in the study. Last year, Josh Gagney and colleagues published a paper that showed how the Sentinel standardized modular programs were able to successfully replicate the earlier study. And they were able to basically use the same data and the same kind of study design to look at how well they could produce the same answers from this study. And in their paper, they show that table 3, the incidence rate was 4.56. 4.56 is pretty close to 4.38. I think intuitively I'd say, yeah, I feel pretty good that they've gotten that pretty close. Here's the problem with big data. What I'm showing you in blue is that first paper, the Toe et al. Estimate of 4.38. And the yellow, I'm showing you Josh Gagne's confidence interval and estimate. And what you see is that these two probability distributions, they only overlap 19%, meaning that the paper is a demonstration of the successful reproduction of two studies, and yet we're saying there's an 81% chance that it's actually a different number. Now here, I would actually assert that their study is a very nice demonstration, but we might actually have the wrong statistics we're using to infer this, because 4.38 and 4.55 or whatever, those are pretty close numbers. But we as a community haven't gotten to grips with how to communicate uncertainty. We're using sampling statistics from unbiased estimers predicated in the history of randomized clinical trial data, and then we're going banging them on big data sets and assuming we're going to get similar things to interpret. Are we downhearted yet? All right, so let's talk about a known unknown. I don't know how many people saw this paper. This came out a couple months ago. In BMJ, it said that the risk of death, there's a risk of death among users of proton pump inhibitors. Show your hands if anybody saw this. First, did anybody see it from the published literature? One sheepish hand. It's okay, it won't bite. How many people saw this from the mainstream media? Okay, we see like 7 or 8 hands where they saw it from the mainstream media. I came to know of this because my dad called me up and said, should I stop taking my drug? So let's take an examination. So they took the VA population and they looked at whether or not PPIs increased the risk of death. And specifically, they assert that this is a known unknown because they say, however, whether PPI use is associated with excess death is unknown. But now they're giving us the answer. They're providing us the known unknown. And to do that, they give us an estimate in the abstract that says the risk of death was increased when considering PPI use versus no PPI use, with a hazard ratio of 1.15 and a confidence interval from 1.14 to 1.15. So either we got to petition the editors to allow us to go to like 7 significant digits in our journal publications, or we're probably fooling ourselves with the precision that these studies are providing. Now, the thing is, this research group is a really good research group. They did perfectly correct statistics the way that we were all been classically taught. In fact, they did something pretty remarkable. They did a whole bunch of sensitivity analysis, because I'm sure the reviewers didn't really buy this. I'm going to just zoom in on 4 of the estimates that are specifically trying to compare PPI use versus H2 blocker use, just to try to see if we can summarize this point a little bit better. And I'm just going to draw it because it might be a little bit easier to see. So that first estimate up top is their primary analysis. It says that the point estimate, the hazard ratio is 1.25 with a confidence interval from 1.23 to 1.28. But they then performed a series of sensitivity analysis, like they used the high dimensional propensity score approach that Schneeweiss et al. Have advocated. That produces a little bit lower estimate. They did time-dependent propensity score matching. That produced a higher estimate. You can see collectively here, if you just look at the summary bounds, kind of like what UpToDate did, it might be more plausible to say that the estimate's somewhere between 1.13 and 1.39, because that covers the whole space, than it is to assert I had a primary analysis, and therefore this number, 1.25 with a 1.23 to 1.28, is correct. Because after all, if you look at that, most of the estimates from the sensitivity analysis don't even fall within that range at all. The point here is that we're dealing with a lot of systematic error, not random variation, and yet we're using traditional statistics to try to make us think about that. So if one of the things we should be thinking about is how do we provide credible intervals that actually express our uncertainty in legitimate terms rather than in the statistical artifacts that are probably misleading us. And I mentioned that my dad contacted me about this, and it was because of this article. It was in CBS News that said some heartburn drugs are linked to a higher risk of death. And I was like, well, I'm sure nobody else picked it up. But then CNN, as soon as I clicked on it, it said heartburn drugs are tied to an increased risk of early death. And even The Guardian picked it up and said people taking heartburn drugs could have a higher risk of death. Now, if you read any of those titles, I defy you to find where they show the confidence interval and express the uncertainty. I suppose the word linked could be, or tied, maybe you say as long as it's a loose tie, then it's not too definitive. But the problem, that's a very real problem we have, is that we produce statistics that we don't even necessarily believe are credible. These things get picked up and communicated in the mainstream media in a way that potentially could be dangerous. So known unknowns number 2. So the FDA has done something pretty remarkable. They've started to post potential signals of serious risks on their website on a quarterly basis, I think it is. And within the Odyssey community, we're looking at this website because we were interested in what questions the FDA might have. And one of them that piqued somebody's interest was Keppra, which is a drug for epilepsy. It said that the potential risk was angioedema, and it said the FDA is evaluating the need for regulatory action. So we waited a little bit and were wondering if that was going to happen. And then we said, well, you know what? If they're thinking about the need of regulatory action, I wonder if more evidence could be helpful. So we conducted an analysis within the Odyssey community across our voluntary network. We wrote a protocol and placed it in the public domain for everybody to read and see. We placed all of the code to conduct this analysis publicly available on our GitHub repository using standard data quality checked tools so that everybody could see what was happening before it ever happened. We conducted a pretty traditional epidemiologic design. It'll probably be similar to what Jeff will probably talk about in the next presentation. This is a new user cohort design using propensity score matching. We looked at multiple times at risk. We were able to perform a meta-analysis across our results, and we also used negative controls to be able to calibrate whether or not we were seeing residual error. This work started, start of the year, and a few months later, we were able to have the analysis completed and submitted to publication. And here's a little screenshot with, this journal's awesome because they put a headshot of the lead author in there. So I think that was the main reason we had John Duke be the lead author, because he looks way cooler than I would have. So here you can see the risk of angioedema associated with, but here's what I really want to show you is in terms of the results. So the first thing is that we exposed each of the databases that voluntarily contributed to this study. We showed how many people were in those databases and how many events they observed. Together, we had over 55,000 patients in 10 different databases. We also estimated the effect estimate at each of one of those sites, and we were able to show those together. Now, usually we think about big data as compiling data into a common source and one particular repository that we're going to use. But actually here I would actually say that what we did was very different and actually quite more useful. Rather than saying we have one answer that we don't believe this is a relationship, we have 10 answers, and all 10 answers said the rate of angioedema is low and we don't see an increased risk in 10 different sources. And so yes, we have conducted a meta-analysis to pool that together into one number, but I would actually argue that that one number is irrelevant. What's way more powerful about having a network is having 10 answers to the same question. All right, so last thing that I want to highlight is the unknown unknowns. And I'd like to just make an assertion of what could be the opportunity for big data that we can make here. So Don Runsell talks about there's unknown unknowns, and I would actually argue in our drug safety context, what we really want to know is how can we take the things that we're not even looking for right now and bucket them into either things that are known, meaning we know there's a side effect, or we know there's not a side effect, or be clear about the fact that we don't yet know. Put it into the bucket of known unknowns. So recent research done by Voss et al. That was presented at ISPE a few weeks ago, we looked at the published literature to see how often drugs were associated with, how often there was published evidence that suggested either positively, negatively, or equivocal whether or not there was information to link a drug to an outcome. And what this bar chart is actually showing you is some of the key safety outcomes that we typically think about and what percentage of drugs had at least one published article that talked about it. So for example, reading this off, anaphylaxis, 44% of drugs have at least one article that talks about whether or not there's a relationship with anaphylaxis. That means 56% of drugs don't have any evidence one way or the other. Now going back to Rumsfeld quote, we could think about that as the absence of evidence tells us there's nothing going on. But I, for one, would much rather us have evidence out there that says we're not seeing anything. To do that means we need to get proactive and start generating information about all drugs and all outcomes simultaneously and put that information out there, even if we don't quite know how to interpret it. What we've been doing within Odyssey is trying to build a software package that's going to allow us to do that, to run the analysis for every drug and every outcome, using those same epidemiologic best practices and calibration techniques. But the idea is that there should be an open public repository that at least tells us what we know and whether or not we're confident or not. I would assert that that information isn't about doing signal detection or signal evaluation, but this is instead about how do we do a better job of doing clinical characterization, to be honest about what we know about how often things happen, to be honest about our incidence and how frequently side effects happen, and for population level effect estimate, being honest that we are monitoring things that we don't know about and being honest about what we have learned about those things that we did. As it specifically relates to the known unknowns, so often we're doing population level effect estimates on an overall population. We're sitting here at a pediatric research study, pediatric research meeting, where most studies start with an inclusion criteria that says age greater than 18. What if instead we actually allowed all patients into our studies and we specifically and proactively did subgroup analyses to actually say, not let's exclude the kids, but instead let's examine the kids specifically? What if we could make pediatric research either a known unknown or a known known, rather than us currently fussing about where we're going to start to answer the question? So with that, I will stop, and I think we're having a panel after Jeff, so I'll probably just stop now, Ann. Yeah? All right.
Thanks a lot, Patrick. And I think we'll go on to the next person, and then we'll talk in the context of the panel. Okay, we're going to move on. And then after we hear from Dr. Brown, we'll go on to the panel. Dr. Brown is an associate professor in the Department of Population Medicine at Harvard Medical School and the Harvard Pilgrim Health Care Institute. He is going to be talking with us about medical product safety surveillance research in multi-site settings.